very good morning to all of you present, everyone present here. On behalf of NIPER SS Nagar, myself, Dr. Pooja Arora, wish you, a warm on the auspicious occasion of PHARMACON 2022. So we are actually going to start the session number 6 of NIPER PHARMACON 2022, which is challenges in key intermediates and API development. So I kindly request all to please switch off your mobile phones or put them on the silent mode. So today's session will be chaired by Dr. Shashikant Tiwari sir from National the Chem Laboratory and Dr. Jaideep Laha sir from the Department of Pharmaceutical Technology Process Development, NIPER Mohali. So I'll just introduce about the chairperson. He's Dr. Shashikant Tiwari. He's a PhD from National Chemical Laboratory, Pune, one of the prestigious research institutes in India. After completing his PhD, he moved for his postdoc to Germany. And after completing his postdoc, he decided to come back to India and join Chem Biotech Research Private Limited, Pune, as a senior research scientist. He has developed expertise in route scouting, process development using green chemistry concepts, new product development, and technology transfer from the manufacturing of structurally complex APIs, early phase of APIs, and NCE molecules. He's having more than 10 publications in the reputed chemistry journals and more than 15 patents in his credit. He carries 25 years rich experience in chemical research of pharmaceutical importance. This session would be co-chaired by Dr. Jaideep Laha sir. He's a professor in the Department of Pharmaceutical Technology Process Chemistry at NIPER SS Nagar. Prior to he was in the laboratory for drug discovery in neurodegeneration at Harvard Medical School. He's an author and a co-author of over 70 research papers, and his area of research includes, but it is not limited to, organic chemistry of drug degradation and integration, synthesis of heterocycles, and generic APIs. Today he's present with us as a co-chair of this event. So I would request the chairperson and the co-chair to please take their respective seats. Thank you, sir. I would ask the chairperson, please kindly open the technical session 6th. Wait just a second.
Yeah, good morning everybody. So now it's my privilege to introduce Dr. Ketan Agrawal, who is our first speaker for today's session. Dr. Ketan Agrawal is basically from Gujarat and is having 30 years of experience in pharmaceutical industry. Out of 30, last 15 years, he is working with regulatory bodies and he has developed expertise in regulatory intelligence and compliance, and he is engaged with industry associations on OS, QBD. In pharmaceutical industry, OS, QBD is the big things. Day in, day out, we have to work, we need expertise, and risk management plan, technology transfer. So then he also established his company in North America, and the name of RK3 Solution LLC in 2019, and supporting across the industry and providing consultations to pharmaceutical industry. He is also associate engaged with NIPER as a guest faculty for the course of regulatory affairs. Now I invite Dr. Ketan for his lecture.
Morning, everybody. Sorry. I hope you had a good tea today, and I was surprised to have my eye caught red-handed. Supposed to be in the second or third row, but I'm in first now. So, okay. So today we'll be talking about nitrosamine impurities, which is nitrosamine drug-related impurities, drug-related substances. Just give me a second. Okay. So since 2018, pharmaceutical industry particularly was caught red-handed with the nitrosamine impurity, which were known since very long. But in the pharmaceutical world, this was the first time in 2018 we were caught starting with the valsartan, and then so on. Still it is going on very well for the regulators and for the industries and for the chemical and instrument suppliers all. So I'll just share my views on yes, sure. Okay. Hello. Hello. It's okay. Hello. Hello. So this has hugely impacted mainly the research scientists, process scientists. They are forcefully formed to have a new research teams with respect to nitrosamine impurities and all kind of new further process research. So we'll just have a view on the same. Next slide, please. Is any method you have to keep pressing? So this is brief about myself. I am Ketan Agrawat, as Dr. Tiwari has introduced rightly. Thank you very much, sir, for kind words. I have around, I will use this then. Is okay? Yeah, okay. So I'll just have to keep myself steady at one place. So this is brief about myself. I'm Ketan Agrawat, around 30 years of experience in quality and regulatory, mainly in the API and intermediate space. And since last 3 years, I am into consulting mode to do some of my own work in my own field. And we support mainly the small scale and mid scale pharmaceutical players with respect to their greenfield projects, or maybe setting up the QMS and regulatory submission, global regulatory submissions all over the world. Okay. Yeah, next slide, please. So this is mainly my deck of slides today. We'll go into regulatory intelligence, then nitrosamine, few introductions on nitrosamine impurities, Q7, non-nitrosamines, then potential sources, and so on, and then finally the learnings for research professionals, where they should concentrate, how they should concentrate. Yeah, next slide, please. So regulatory intelligence. What is regulatory intelligence? Whenever we have a glass of water or any kind of food or anything, everybody will see first the label, ISI label or brand name or anything. That is where regulatory comes into picture. Everything is certified by a regulatory body, particularly say a particular certain regulatory bodies. Food, we have FSSI. Drug, we have CDSCO and FDA, local FDAs. Banking, we have IRB, RBI, sorry. So on, we have regulatory agencies for almost every processes, every products in for our daily routine. Next slide, please. So these are nitrosamines and NDSRIs, nitrosamine drug-related substances, drug-related impurities, NDSRI. These are mainly the cohort of concern. When I say cohort of concern, the typical TTC approach does not apply here. We have to go as per the toxicological assessment and the dosage of the drug substance, and then it has to be calculated. So for every nitrosamine or cohort of concern impurity, there is a separate limits applied. It's not just like 1.5 microgram per day. It has to be calculated according to the drug substance or drug product dosage form. And EMA has classified this as 18 nanogram per day for all nitrosamines where there is no non-toxicological data available. So blankly you have to apply 18 nanogram per day impurities. Next slide, please. This is general structure. Everybody, I think, should be aware of this by now. It's almost 2.5, 3 years. This is general structure of N-nitrosamines, and the possible formation scheme is here. So N-nitroso, it is called N-nitroso compounds, right? And it's characterized by N double bond O, and they exist in several forms, including symmetrical dialkyl nitrosamines, asymmetrical dialkyl nitrosamines, and functional groups, and so on. It's go on changing from related alkyl groups. So it is ethyl, it is NDEA, it is methyl, it is NDMA, like so on. And most significant source of these compounds are nitration of amine and amide precursors, as reported in various literatures. Next slide, please. From regulatory point of view, one of the major concerns, this is one of the major concerns where the BGI or potential genotoxic impurities are not adequately addressed in the drug master files. Drug master file, or you can say your dossiers, formulation dossiers, wherever drug master files are not available, the necessary information should be given into the dossiers. This is one of the major deficiencies we encounter. And again, the discovery of these has put a lot of pressure on our R&D guys, research guys. And on the other hand, the impurity suppliers and instrument suppliers are more than happy to support you with the supply of impurities, and LCMS, GCMS, whatever instruments are required for the analysis. And this has to be calculated by, as I mentioned earlier, it's 1.5 microgram per day divided by the dose. That is how it has to be calculated. Next slide, please. Now, it started with one impurity in one sartan, and so on, different sartans. Almost all sartans came into picture then. Slowly it has opened a Pandora's box. We have now around more than 42 non-nitrosamines in the knowledge. Starting with all sartans. Ranitidine is completely vanished of the market. Metformin, rifampin, quinapril, orphanadrine, nizatidine. Most recent cases are varenicline and sitagliptin. Sitagliptin just August only they have reported. Varenicline is again going off the market. They have recalled the product. Sitagliptin, because of the major major market, they have not recalled, but there are various investigations going on for the sitagliptin also. So like all sartans, I believe all gliptins will also come into picture now. So the list is just non-list. We have put around 12 nitrosamines. So if you from chemistry point of view, if you read the names, if it is ethyl, it is NDEA. If it is methyl, it is NDMA. So on. Your process, depending upon the solvents used in your process, you have the different impurities. So you have to classify, you have to investigate, and you have to theoretically and then practically you have to prove the absence of these impurities in your drug substance. Right? Next slide. Next slide, please. So it is believed that almost 20% of the approved drugs, small molecule drugs, has kind of vulnerable amines to form various nitrosamines in your drug substance. So when you say 20% is almost multiple or many generic drug substances will come into picture now. These includes the beta blockers, angiotensin enzymes, converting enzymes, SSRIs. So mainly the most daily used drugs will come into picture now. One of the expert, Martin, had said during their discussion with regulatory focus, the initial perception among the experts was that problem would be contained to around 10, 20 or 30 small molecule drugs. But it has opened a long sequence of chains now. And they have miscalculated or they have misjudged the origin of these nitrosamine impurities. And they did not realize how broad this problem is and how quickly this would become a broad issue among the pharmaceutical industry. Next slide, please. So, this is not, nitrosamines are not new in the chemistry world. It may be a new for pharmaceutical world practically. Theoretically, we all know that there are kind of nitrosamines, but since 2018, this came into picture. So when I say this is very well known in the literature and chemistry, this has become available in a wide variety of foods. For example, meat, bacon, vegetables, dairy products, drinking water. Very well known limits are given in the drinking water specifications. Manufactured and natural products such as agricultural chemicals, tobacco, detergents, solvents, drugs, plastics, cosmetics, etc. Everyone is exposed to some level of nitrosamines at any given point of time. Human exposure to nitrosamine can result from formation of N-nitroso compounds either in food, even during storage or preparation in vivo. Then again, this is probably the first time we have as a pharma industry, we are seeing the practical implications of detection of nitrosamine impurities. Right? Next slide, please. So these are the potential sources, very well discussed in the literature, very well known since last 2.5 years and this should be known to all pharmaceutical scientists developing generic or developing new innovative drugs. So main basic criteria is sodium nitrite or other nitrosetting agent in the presence of secondary or tertiary amine, quaternary ammonium salt, with certain process criteria, process characteristics. Use of these nitrosetting agents and amines along with the reagent solvents catalyst, which are susceptible to degradation to secondary or tertiary amine within the same and different process steps. Okay. Use of contaminated raw material sources, recovered solvents, reagents, catalyst, key starting materials. Use of recovered materials, solvents, reagents, process, catalyst, which are very well defined in our process chemistry, including recovered sources outsourced to third parties who are not aware of the content of nitrosamine contamination. Use of contaminated starting materials and intermediates supplied by vendors that use processes and raw materials which may allow nitrosamine formation. Cross-contamination due to different processes run on the same line. Okay. On the same line due to operator-related errors such as inadequate phase separations. Then the degradation of the process starting materials, intermediate drug substances, including those induced by inherent reactivity. So these are the common sources identified as per the submissions of various drug substance manufacturers, drug product manufacturers. These are not only the sources. There are various other multiple sources which will become known in during the next I think few months or so. So every day, every month you have a new source of nitrosamine impurity depending upon the drug substance or drug product processes. Next substance. Contamination and or trace amount of nitrites in raw materials like water, acid, where you have to have risk assessment for nitrites, trace amount of nitrites in your drug substance, drug product processes, including excipients. Unexpected source of contamination is your drug product packaging material, which contains several amines into it, printing ink, which may contain amine. So that reacts with the drug substance or drug product packed into those material, and then they contaminate. They form the nitrosamine and they contaminate. We have n number of nitrosamines. We are talking about nitrosamine, but there are differences in nitrosamines and nitrosamides both. Both are different in genotoxicity evaluations. They are primary, and nitroso is derivative of primary amides, and alkyl urea, guanidine, carbamates. They are not in the same category as nitrosamines, which are again nitroso derivative of amines. Okay, next slide. So this table gives you an idea of how nitrosamines perform and nitrosamide perform in the genotoxicity evaluation. Nitrosamine will give you the almost similar potency, but they are not in the cohort of concern. Nitrosamides are considered as a cause of concern, but nitrosamides are considered as the normal genotoxic impurities. Guanidines, again, we have different classes. nitrosocarbamate, we have different classes. Nitrosourea, we have different perspective on those, both the impurities. Okay, next slide. So as a process chemist, as a process researcher, we do have to have a perfect literature search for carcinogenicity studies for each compound. Different compounds have different mutagenicity, different carcinogenicity. Both compounds will vary from carcinogenicity or mutagenicity perspectives. For example, I have given few examples here. Piperazines, they behave differently. The one is, I'm not process chemist, so I may not pronounce perfectly. So red ones are mutagenic, red ones have mutagenic potential. Green ones doesn't have, they are considered as a non-mutagenic compounds. Okay. Piperidines, again, they have both types of compounds. Dialkyl nitrosamines, again, they have, depending upon different groups, they behave differently with the in the mutagenicity. Next slide. So as a process chemist, we should look for new chemistry leading to lesser impurity profile in starting materials, intermediates, and your finished product, drug substance or drug product. Always perfect your chemistry where you have lesser impurity profile. Priority should be given to alternate processes, circumventing the PGI formation instead of increasing yield, instead of commercial gains, which has happened in the certain cases where they have given preference to the yield instead of the formation of nitrosamine impurity, which became known later on, but that is a lesson from the valsartan case. Strive for deeper understanding of existing reactions and process routes to be pursued to mitigate the PGI presence, potential genotoxic impurity presence. Extensive literature review. We have thousands of articles, literatures available now only for nitrosamine. So do your perfect literature search and try to mitigate the PGI impurity formations. Perfect your chemistry, generation, purge factor, control strategies, quality by design principles, control by design principles, quality by design, control by design. Every time you cannot have controls at the last stage of drug substance formation. You have to start from the very beginning, starting of your key starting material or raw material or intermediate. Everywhere you need to put controls. Then only you can have the defined quality of your drug substances. Vigorous evaluation of your supply chain materials, supply chain partners. Sit with your partners, discuss with your partners what you want, how they can give. If they cannot give, probably you can support them with your chemistry knowledge, with your practical knowledge, with your process knowledge. Sit with your supply chain partners and discuss with them what you want, how they should mitigate the PGI risk. There are various examples of drug substance, drug products going off the market because of supply chain issues, because of the vendor approval, because of the your partner could not control or your partner could not have the adequate specification for the same. You should have a cross-functional team for the review of a technical review of your manufacturing process. You need R&D person, you need quality assurance person, you need quality control person, you need process engineers, you need production persons. Everybody should come on board to review your process so that you have enough control, so that you have designed controls on your drug substance, drug product specifications. Okay. And last but not the least, it's okay to keep a tab on company business goal profits, but keep it or do it within regulatory framework. Do not ignore the regulatory guidances. You do not ignore the design principles. Do not ignore the controlled principles. Always, company is meant for profitable businesses, right? But we do have our maybe we can say ethics to keep it within the regulatory guidelines. Okay. Next slide, please. So, in-depth understanding of formation, fat, and purge of all impurities will facilitate the selection of appropriate control strategy and will speed up the approval of applications. If you do a literature search, there are number of applications, number of innovator drugs which may FDA has rejected due to the process issues, due to the chemistry issues, due to the impurity issues. So, enough understanding of your route of synthesis, enough understanding of your impurity profile and the presentable material or presentable documentation to regulatory authority will speed up your approvals. Okay. So, you need a global perspective, critical thinking skills, lifelong learning. So, you can apply certain learnings to the other route of synthesis, other processes. Okay, global perspective. Nowadays all the drugs are going globally. You are not limiting yourself to say US market or Canada market or Europe market. So you have to keep all regulatory guidelines into focus, and then you have to design your route of synthesis. You have to design your regulatory strategies. You have to design your drug substance specifications. In one of the slides, when I say EMA has put 18 nanogram per day of general PGIs where there is no toxicological data available. So you have to keep those guidelines applicable for your US market products also, where you have stringent criteria and you comply with the global regulatory specifications. So as Benjamin Franklin said, an ounce of prevention is worth a pound of cure, right? So in the beginning, if you keep a tab on the regulatory strategies, drug substance specifications, stringent drug substance specifications, you are going to get it. You are going to get a faster approval of your drug product drug applications and drug substance application, drug master files, whatever the regulatory submissions you have. Impact, potential impact. If you are not doing this, maybe a product recall. If you see the history of product recalls in last 2 to 3 years, you have, I think, highest number of recalls due to nitrosamine impurities, right? They on the paper or on the website you may see manufacturing issues, written as manufacturing issues, but basically they are mainly with respect to the nitrosamine issues. All sartans, ranitidine, metformin, all other drugs which I listed there, sitagliptin including. So they are known for the nitrosamine related issues and they are recalled for that. Back on the strategy table. Yeah. Overhauling of your supply chain issues, withdrawal of regulatory submissions. Many DMF holders have withdrawn their submission of valsartan, irbesartan, losartan, all these things. Production stops, the warning letters, reputation damage, and financial losses. This is the impact of your not going as per the regulatory guidelines. Yeah. Next slide. So for risk assessment, assess the risk. Confirmatory testing, you have to go both ways, theoretical versus practical. Theoretically, you prove you don't have any nitrosamines, and then practically prove you really don't have any nitrosamine. Risk assessment, you prepare a risk assessment report. Based on risk assessment, you design your process, you optimize your process, mitigate the risk. That is your design controls. Yeah. Last, I have just kept a few literatures, important literatures based on my understanding of the nitrosamine issues. We have a excellent article with the Andrew Tesdal, which is very well known figure in PGI for the PGI impurities. So if you can get a chance, please get that book and review. Another important issue, the link would be the USP has formed a nitrosamine forum where you can get a login and get the discussions going on for various number of impurities. Those are the, and of course EDQM and USP has started the general chapters on nitrosamines. Okay. Okay. Thank you. Next slide. Thank you.
Thank you, Dr. Ketan, for nice introductory session on nitrosamine impurities,
Thank you.
which is a really burning issue for pharmaceutical industry.
Certainly.
And today I would state one thing in your lecture. When we are developing a pharmaceutical product, as a process chemist or regulatory, our first approach should be patient first. If you think patient first, then all things will come. If you think profit first, then all things will come. So now floor is open for question answer session. We have a question, we can have a quick 3 to 4 question from the audience.
Yes, please.
Yeah, so I have one question. What is the Indian standards? Means in India there is no regulatory on NDSRI or neither on the nitrosamine. So do you say that it will come to a Indian regulatory on DCGI or a US, means like a US FDA or others?
It has already come. IP has recently, I think last week also, they published a general chapter on nitrosamine.
True, but for an approval process, still is there a requirement from our Indian FDA to see on a risk assessment and others, or it is like only the IP general chapter which doesn't speak about any of the monographs?
Correct, but when you apply for the drug approval of your drug substance or drug product, you may have to comply with the general chapter requirements also.
Okay.
As per that, you may have to submit the different risk assessment reports.
True.
You have to comply with the. Again, this is, see, authorities going as per the industry knowledge. Authority doesn't have their own set of knowledge or laboratory or whatever it is. They are going as per the industry knowledge. Whatever industry submits, based on that, they prepare a guideline, they prepare certain limit, certain specifications also. If you see nitrosamine in 2018, there were no
Nothing.
monographs, there were no general chapters. But when industry submitted, starting with the Chinese guy who has submitted the unknown impurity formation, they immediately within I think 6 or 8 months or so, we have USP general chapter for nitrosamine, we have EDQM general chapter for nitrosamine. Then US FDA has published a guide, very well guideline on nitrosamine. So in my previous slide also I said, think on the global perspective. So don't limit yourself to Indian specification or Indian approval.
True.
As a manufacturer, I should be proactive on
Submitting.
doing these things.
Yeah, so the another question I have, that the 18 nanogram per day, that limit is not fixed for all the drugs. Means US FDA comes back and tell that for this it is like 1000 nanogram per day. So the LASA and other teams working with the EMA for having a AI based or the software based understanding, because nobody is doing the animal toxicity for all, and it's not possible. So what are your thoughts on that? Means, do we have anything indigenous where we can do a predictions based on the metabolism potentials, or some type of a kinetics, and can predict and submit the data, or still we have to follow the 18 nanogram?
See, that is the EMA has published that 18 nanogram for the impurities where you don't have any toxicological data. So it is always better to work with authority based on your report, based on your documents. You submit the documents and you work with authorities. That is the approach I will take. I will prefer to take that approach because still authority doesn't have full idea of how nitrosamines are generated, how they are controlled. Okay, so it is always better to work with authorities. I will suggest you submit your data, whatever data you have, and then you work with them.
Thanks. Thanks.
Thank you.
Any other question from audience?
Yeah.
If there is no question, then we'll discuss.
Thanks so much, sir, for sharing the knowledge on regulatory intelligence. I think that's a need of an hour now. We all have to work on it. So, may I please kindly request chairperson to please, hand over the memento to sir. Thank you, sir. Thanks.
Yes, okay. Now we have next speaker, Dr. Andiyappan Murugan. He is basically from Tamil Nadu and currently working as a Vice President R&D, Troika Pharma, Ahmedabad. He is a PhD from National Chemical Laboratory and 2-year postdoc from US. Followed by, he has worked companies like AstraZeneca, Dr. Reddy, and having total 20 year experience in pharmaceutical industry at various level. His expertise is route designing, process safety, and as a very strong scientific and leadership talent. So let's, on behalf of audience, I would like to invite Dr. Murugan for his lecture on the design principle of the API process development. Please.
Thank you, Tiwariji, for your kind introduction. I also thank the organizers for the invitation. And it's good to be here, first time in Chandigarh. I looked very, very different. So it's a beautiful city. And yeah, so today my topic of the talk is design principles of API process development. So API is the active part of any drug, and then it's also source of all the impurities and the costs and all the implications. So I thought that okay, let me share some of the concepts that I feel that most important, and while we embark upon developing a process for API. So let's look at making a medicine, applying QBD concept. How hard can it be, right? So let's just simplify QBD development or QBD principles. Okay, so let's consider making a pizza. I think pizza is such an important food nowadays, at least for this generation. So let's see, like if you do a retrosynthesis of pizza, so the pizza, so for a for a consumer or a customer, so what we call it as QTPP, quality target product profile. That's for the medicine. So what should be the QTPP of pizza? You consider flavor, taste, nutrition values, appearance, right? So to achieve that, so you need to have certain critical quality attributes, being measurable quality attributes, that if you ensure that those properties are met, then you have the right quality pizza. So for that, so you need raw materials. So you have a bread, cheese, and the toppings, and they in fact, sorry, yeah, so in turn their source from the bread comes from the egg and the wheat flour, and wheat source from wheat and chicken, and cheese comes from the milk and the cow, and the same to do for the meat. Now, to achieve the right quality pizza, so you need to control qualities every stages with the right specifications. And also the process is converting milk into cheese and cheese into thing. So even you control everything, and if you don't bake it properly, then also you mess up the pizza. So baking is the process. Like you have the process control, the temperature, time, etc., etc. That's what we call it. So this is we call it as quality by design. You design specification, design the process at every stage to ensure a reliable end product. Okay. So what does it look for API? Very similar. Okay. You have start with chemical component, couple of chemical components, put them together, do some functional group interconversion, then maybe an activator, introduce another component here, and then another couple of chemical transformations, and then introduce another chemical component, and then same way, maybe if it's a convergent synthesis, you have one more starting material, few conversions, you have another starting material B, and then put them together to get the API. Same, you control the qualities and processes at every stage, and you will get a reliable process for this. So to move on, so when you embark on process development on an API, so there are certain goals to your development. The goals are summarized as SELECT, we call. So this is the SELECT principles, summarized in this chemical review by a few of my pharma colleagues. Okay, and if anybody reading process chemistry, please do read this review. It's very, very informative. It almost summarizes every aspect of process chemistry that is involved. So select safety of the process, safety of the material that you handle, and establish the control. Environment, look at reducing the effluent generations, increase the process mass intensity. Legal, look for the freedom to operate or have innovative process. Economy again, you have to have the right yield and right cost, and control over the qualities. And then finally throughput also comes from you should have a high right yield and then right processing times to achieve. So this is basically the target for any process chemist who takes up the process development of an API. Okay, put those things into a QBD framework and put it as a plan. So we have various stages of process development. Is starting from once you have given the molecule, you go see the route, look for various synthetic routes and select the most suitable route, and then go for stepwise screening of reagents, solvents, and then optimize your process parameters, and then study the scale-up parameters, scale-dependent, scale-independent parameters, and then once you have, and then establish the design space, then go for the trial and validation. Okay, and then once your trial and validation done and stability done, go for a DMF filing or a NDA filing. So this is the general product life cycle of any product development. So when we look at stepwise, the route selection, you first understand the QTPP and CQAs of the API that you develop, and then accordingly devise your route of synthesis which should yield the right quality API reliably, and then do screen for the different reagents and solvents to choose most suitable reagent. I will come in a moment and what it means to choose a reagent and solvent, and the process optimization. So you know that stoichiometries, solvent volumes and temperature, time, etc., etc. Those are all constitute into process parameters, and then identify the critical process parameter by means of the process risk assessment, DOE, CPP, CMA, mapping with CQAs, and then establish a design space. And these are all the critical components of your development, and the QBD is no different. Even if you look at many industries, follow almost everything same. The only thing is you don't have a structured way of planned way of development. That's all. But more or less everyone follows this to achieve the right quality. Right? That's that by default it happens. So what we are saying is QBD is just at the start of development itself have a plan, so you are there. So moving on, so let's take the first one. You want to do a reagent selection, how a reagent should be. Like for any chemical transformation, your guys as students are there. If I say amide, you can list 10 different ways of making an amide. But for a given structure, what should be the most appropriate reagent, right? So your reagent should be selected in such a way that it is specific for the desired transformation. Okay, and it doesn't touch other functional groups in the molecule. It doesn't react with other reagents or solvents in the reaction. And if it is regioselective, it should be regioselective. If applicable, it should also be stereoselective. And it should, the reaction should facilitate, the reagent should facilitate the reaction under ambient temperature. I don't want minus 78 degree. I don't want plus 200 degree. Anything around room temperature will be most desirable, and also reacts at the reasonable rate. Different reagents can give at the different rate of reaction. I don't want to run the reaction for 3-4 days and occupy my plant, which will impact the throughput. And same way, it should not be toxic or the least toxic, and shouldn't produce toxic waste, and should be commercially available, and wherever possible, if it is catalytic, look for the catalyst which can be recycled and reused. And similar thing for the solvent selection, should be commercially available, give this safe scalable process, and doesn't interfere with the reaction, rather instead it facilitates the reaction, and also helps in isolating the product in pure form or by the way of crystallization, and it doesn't contaminate the intermediate or API, and easily removed. So these are the the considerations you put in when you choose a solvent for a particular transformation, right? So let me kind of illustrate those with a few examples, and maybe I will give examples and for a different concept. So this one for the good route design, and SAT3264 is the product which was being developed for COPD asthma, targeting IKK2 pathway, and this is the structure of the product. And we were involved in the late stage chemical development, so we generally get a route of synthesis from medicinal chemistry, and so this was the route given. So if you look at the structure, you have a central aromatic ring, and the 3 rings are connected together. So this is how I would look at the disconnection. So and the same thing was done. So first that this bromofluorobenzene was reacted with this pyrrolidinol, and get by SNAR you get this one, and then halogen metal exchange followed by boronic acid. So you get this boronic acid, and this boronic acid undergoes Suzuki with this bromothiophene. You get this component, and then debug it and brominate it, because I need to put one more ring here. So bromination. So this was a kind of non-selective bromination had other impurities also, bromination over here and over here also, and then final Suzuki to get this on. But then yes, we did deliver product for the toxicological studies, but it did have its own issues. First thing is poor yield due to selectivity issues at the bromination step, and also created corresponding impurities. And the API was very insoluble in most solvents, and we had a Suzuki reaction at the last step, so there was a palladium contamination in the product, and more so that it was a free secondary amine and had an affinity towards palladium, which was difficult to remove. And then there was a crystallization in DMSO, and then DMSO was also contaminating the product in the form of solvate. So after all this assessment, then we felt that when we did the selective assessment, this is what happened. We didn't have good safety profile, though acceptable safety profile in the sense the process safety. And environment, there was a lot of waste generation due to poor yields and very less control over the quality of the product, and poor yield also lead to a very low throughput. So when we embarked upon new route, so what we felt after all this assessment and the way considering the nature of the molecule, we felt that may not be the right route that we are following. So we may have to come up with another suitable route which can circumvent all the problems. So, and also the next phase was for clinical, so we can't have a very high residual palladium or DMSO. So the objectives were like achieve the product with palladium content less than 75 ppm, avoid using DMSO or any high boiling solvent towards same, so that you avoid the solvate formation. And we should also match the impurity profile because we were already completed the tox study, so you cannot introduce any new impurities later on. So based on that, we have devised the strategy. So we thought, so the strategy objective was have the palladium scavenging done at earlier phases, avoid using any high boiling solvents towards end. So again, look at the molecule. So this is the central ring, and then you can disconnect in these three fashions. And what we came out with was with a central ring with the differentially substituted phenyl ring. Okay, and when we disconnect, these are the options we get. So we explored all these options. Finally, came up that so this was the most suitable starting material to start with, and so this is what the route like this. So we kind of rearrange the route of synthesis. So we have now we have started with bromo, fluoro, iodobenzene, where this was generated later in the previous synthesis, but in our synthesis we start with the already commercially available material, and then we give the SNAR, and then you get this bromo. So it's one step. You can complete the Suzuki also in the same part, so you don't have to isolate this one, and then have this halogen metal exchange done. Boronic acid, again the boronic acid can directly be coupled with bromothiophene in the same part, so you get the API. It's just an N-Bach protected API. Now it is N-Bach protected API, so the affinity issue with palladium is removed because palladium, the nitrogen is protected here. At this point, you do the scavenging to remove all the palladiums, and then do the debug and purification. You get the highly pure API. And what we achieved high overall yield, around 34% for 5 stages, and no DMSO or any high boiling solvent, and had less than 10 ppm of palladium. And after that, when we see the select, so we see that we have ticked almost all the boxes. And so that was one. So the other thing I also wanted to speak about is the use of physicochemical properties of your product to devise a suitable process for, say, if you want to extract or purify. These are all very important things when you really want to develop a high quality product. So pKa is one of the things that because most of your APIs and intermediate are either amine based or a carboxylic acid based. So pKa is where you can either protonate your amine to form a salt which will make your product water soluble, or you deprotonate acid, which will create a salt, again can be water soluble. And then you can do vice versa. So the salt can be made free base, and the free bases become organic soluble, and the salts become aqueous soluble. You can make use of these properties to devise a suitable extraction process or a salt formation process. And also this concept can also be used to choose a right base, because if I want to use an amine base in a reaction, I have 20 different options. Okay, so if I know the characteristic of the amines, then and characteristic of my reaction, so I would be precise with 2 or 3 bases which I need to really experiment. I don't have to experiment with all sorts of bases. So that's one important thing. So here, just I wanted to illustrate like one success that we have achieved by using this pKa values. So for one of our products, so it's a very large product molecule, so that's why I just put the most active part involved in the chemical reaction. So the reaction is like the product is this amine is what our target. It was an intermediate basically, and so the synthesis starts with this bromo compound. We do Heck reaction and hydrogenation, and then there is a Hoffman rearrangement to get this primary amine. And during the Heck reaction, we also get some amount of 5 to 7% of this bromo impurity. Now, if you find these two, both are basic, and these two are not solid. This is a gummy mass. And then, so if you want to purify this, then what you do? Do a column chromatography. Can I do column chromatography for 500 kg of material? Not possible. So I need to look at a process by which I can remove this impurity and get pure required intermediate. So what we look at it? So the pKa of this amine is around 10.6 and this amine is around 8.4. So when you do a speciation for this is the speciation for the the intermediate, the required product. You know that at 9 pH and 97% of the product exists in salt form, which means that it will be in aqueous phase. And at pH 9, 80% of the impurity will be in a free base form, which can be extracted into organic phase. So that's what we have done. So taken this crude and dissolved in sulfuric acid and adjusted pH to 9 to 9.5 by dilute aqueous sodium hydroxide, and then extracted the aqueous system with toluene to remove this impurity. Then afterwards again further basification and then extracted into 2-methyl THF, and then we added HCl gas to precipitate the product with greater than 99% purity. Okay, so this is how you need to develop, devise a process so that you exclude the impurities. So this is what's important here is understand the physicochemical properties of the product, then devise the process. Otherwise, yes, you will be able to do a reaction successfully. 5% of a desbromo impurity in Heck reaction is not bad. I still get 90 plus yield. But removing that 5% from the product is tough. And in that process, we may lose 20-30% of the product. So that's where understanding the physicochemical properties of the product is so important, and so you devise the process accordingly. Okay, and then so this is what I basically summarized it. You can use the pKa to devise your process, devise the extraction procedures, devise a good reactions as a reagent selection also it can help. So the last piece I want to talk about is this asymmetry. So one of the successful products from my laboratory. So we have developed this from lead optimization to all the way up to the launch. Okay, and then it was called AZD 9291, and so this is the the product profile when we took up this product. And so on the mice model, so this is for EGFR, non-small cell lung cancer. Okay, and so this is the treatment with the gefitinib, and this is the treatment with our AZD 9291 in mice model. You can see that on 40 days treatment, so the tumor is almost nil, and then it continued to be nil for 100 days. And this is one of the most efficient treatment available for T790M mutations. And this mutation is found almost in now close to 20% of the non-small cell lung cancer population. And it's one of the best treatment available today now, right? Again, so we picked, So we picked up this from the Medicam route, and then we further devised, and so we actually changed the route, and I'm not bringing that. I wanted to talk another some specific aspect here. So this is how the route look like. So the product is a monomethylate salt, and then we start here and do this filter crops with dichloroperimidine, and then SNAR to remove this, and then reduce the introduce this trimethylethylenediamine, and then reduce the nitro, and introduce this 3-chloropropenyl chloride to generate actually an acrylyl function. Here it is important thing is that if you want to create acrylyl amide here, you can use acrylyl chloride, which is also commercially available. But the thing here is acrylyl chloride contains a lot of oligomers and polymeric impurities. They all react, and if you look at the HPLC of this reaction with acrylyl chloride, it's like a Himalayas. You see lots of peaks because of the oligomeric and polymeric peak. So to avoid that, circumvent that, so we had used this as a precursor for the acrylamide. So first do the amide linking, and then do the beta elimination with the triethylamine and solvent, and you get a very clean product here. So everything went well, and this is what was dosed for the all the clinical talks and GLP talks, everything. But for the clinical phase, we wanted to have this monomethylate salt in tablet form. So for clinical phase, we did this. And in the lab, so the addition of methane sulfonic acid in acetone water mixture, we achieved this monomethylate salt. So only critical factor here is, in the absence of water, if I do in acetone, I will get the mixture of salt. So what happens, this is also susceptible for formation of disalt. So when I do it in a non-aqueous system, and I get mixture of free waste, the monosalt, disalt, so I don't get a single product. To get a single product, I need some amount of water. Everything went well. So when we did this on scale, we found some dimer forming, and which resulted in quality failure. So we need to again rebasify it, again resalt it, and we supplied for the phase 1. But then we know that this is not the best process to follow for the future supplies. And this was a most accelerated program then, because it was one of those molecules which were given fast track therapy designation at the beginning, because FDA was giving maybe in early 2010s, they started giving the breakthrough therapy designation and also the fast track. And so this was one of those molecules. And then we had a really good opportunity to fast track this development. Okay, so it was important that API supply shouldn't come on the way of your development. So when we just looked at this and how to develop this, because in the last steps, the purification really consumed a lot of pure material. We are kind of losing 30-40% of the product at the end. So we thought we need to do something about it. So what we did, and we did the risk assessment with mapping the CMAs, CPPs, and the API CQs, and we did come up with a DOE design using this response phase methodology, and then we identified to identify critical process parameter. So what we identified was the water content is the critical process parameter here, because lower water will result in different polymorph, and higher water results in the impurity. So, and then having found that water content is an important factor, then we did do the FMEA process risk assessment, and what we will do to control this, because at the end of DOE we didn't really see any difference in the process. Basic process remains same. So we were still looking at what would have gone wrong in the phase 1 manufacture. So actually what went wrong was after all this, it was only a simple trick in the operation which was required. Okay, so this is the GMP. It's the last step. So you need to do a a candle filtration to remove all the extraneous matters. So remember, this is an acetone water system. So when we were transferring from one reactor to another by using vacuum, and it sucked some amount of acetone into vacuum. So what happened was it changed the ratio of water acetone. So after all this assessment, what we did was in the next campaign, nothing. Instead of applying vacuum into the reservoir, you apply pressure in the source. So you will avoid acetone. This is all was required, and then you have a successful process. Okay, but to arrive at that simple trick, so the process you go through, ensure that okay, if I do this, I'll be able to convince the team that yes, I will be able to produce this product with a reliable quality and yield. So this all was required, and this is big success story for us. And so this is one of those first products where we entered into first time in 2013 and got approval in 2015, less than 2 years. And 2014 we were running phase 1, phase 2, phase 3 simultaneously. And because at that time we were looking at the terminally ill population, so there was never issue of recruiting patients. So whoever gone through the regular treatment and the relapsed, and they were all eligible for this program, and we were able to recruit the patients randomly and rapidly, and could get the approval in 2 years. And today, as we speak, it's generating more than $5 billion for the revenue. And this is actually the revival story of AstraZeneca. Otherwise, Pfizer would have swallowed AstraZeneca by now. And finally, I want to thank all my collaborators, mean that past and present, and whoever, they all have contributed to this work and some of the other ways, and there's so large number, that's why I just put common. And thank you all.
Yeah, thank you, Dr. Murugan, for delivering very nice talk on process design and process design, applying control, QBD. So from his lecture, it is very clear each and every operation in a process chemistry, even in your synthesis when you are doing in lab, very important to observe. Then only you can solve bigger problem. It's more observation only. Observation, observation, observation in experimental science. So now floor is open for question answer. And
Yes.
yeah, go ahead.
So, sir, you say you avoid DMSO, but some reaction is very specific. So, we cannot avoid it. If suppose I take some copper catalyzed reaction, DMSO we have to use because it is the best yield. So, how, because it will always take some compound when you do water workup, how you give any solution to that?
Yeah, so avoid DMSO, this is in the context of this particular product. Again, the very basis of my talk is trying to understand the behavior of the product. Accordingly, you choose the solvent. If my product is susceptible for solvate formation, then whichever solvent which will form the solvate, I may have to avoid it.
No,
Unless
that
it
is
is
fine.
decided.
No, my point is I have to take DMSO now. I don't have option because you getting best yield. So how, because when you do water workup along with DMSO, a lot of yield loss will happen. So you do you have any, because you must have faced similar challenge.
Yes. See, again, DMS is one of the very high profile or highly desired solvents for many organic transformations, no doubt about it. But having said that, if you look at what is DMSO is special about, DMSO is an non-polar solvent with a very high dielectric constant, this property makes sure that many of the most organic components dissolve in the DMSO. I can give you an alternate. I had maybe in another presentation I could have. I can devise the solvent system which can be equivalent to DMSO. Very simple. Take toluene, add some tetrabutyl ammonium chloride. The mixture will give you a very high dielectric constant. Or people do that. Take toluene, put little bit of DMF. You can perform most reactions. So most reactions which can be done in DCM can be done in the mixture of just 5% DMF in toluene. So likewise, you can look for the solvent or solvent combinations which can replace DMSO. But that is if it is necessary. It's not in all cases you need to replace DMSO. DMSO is one of the good solvents.
So, like DMSO, your question is, it is you are getting lot of yield loss, but you have to optimize your water ratio, how much you are using. So, neutralize the polarity of DMSO.
If I optimize, TMSO will come with my product. That
No.
is another hard. Okay, I will not go there. It is more deep.
Yeah, so it's like this. It's not the additional water is not the only way to isolate the product from DMSO. You can also consider anti-solvent, non-polar solvent.
So, sir, I go next question. You say you by PK calculation, so you predict by software or you do experimental for each product?
For this, we have only used the software. Okay, but you can do experimentally. You can calculate the
So,
measure the PK.
what software you are using for that? Any
I think
specific?
it was done in ACDA.
ACD. So, 2-methyl THF you have used specifically in one case. So, is there any specific advantage?
Yeah, so it's an ether solvent, and 2-methyl THF, it's having advantages over THF in terms of amount of peroxide form. So it's a safer process compared to THF. Not all safe, but it's much better than THF.
Dr. Murugan, it was a wonderful presentation. So we enjoyed your talk. Basically, I wish to understand that what Dr. Swain raised, kinetics data, how it influences the efficiency of the process.
Yeah. So kinetics data you evaluate in two ways. One is how fast you can complete your reaction, because first understand whether it follows a zero order reaction or a first order reaction or a second order reaction. Okay, if you know if it is a second order reaction, at some point the reaction will slow down, like the rate of reaction will fall like this. Okay, so you need to understand what to do if it is a second order kinetics, then you need to understand what. That's one. Other thing is what should be the optimal rate. Sometimes if I want to accelerate the reaction, I may end up producing other impurities. So it is important to study at different intervals what my reaction profile is, and in terms of conversion and also in terms of impurity formation. And almost all reaction you will find, if I get a faster kinetics, invariably there will be some impurity coming in. So I need, sometimes I may have to do something to slow down the kinetics also. So, both the possibilities. So you need to see again, substrate specific, you need to see what. Yeah.
Comparatively, don't you get mixture of these products? And specifically, you are mentioning that fluoride
Yeah. So let's first understand the reactivity pattern here, right? So fluoro, you don't break this bond by in terms of insertion. Bromo and iodo, you do insertion. Like when I do a palladium coupling, what happens is that palladium gets inserted into the bromo and your carbon. But in this case, what I am trying to do is the nucleophilic displacement. So, anion generates here, then it displaces here. Okay, fluorine is susceptible for displacement reaction. You cannot, otherwise you cannot break this bond to introduce the palladium or any metal here. So what you are looking at here is the way of displacing it. Okay, here I am trying to do a nucleophilic displacement. And bromine, carbon bromine bond is not known to be, at least in the aromatic ring, not known to be susceptible for nucleophilic separation. Okay, and fluorine is susceptible for nucleophilic separation. That exactly we are trying to do here. The higher order fluorine, or if you move from fluorine to iodine, okay, the nucleophilic displacement capability reduces, but the metal introduction or the metal insertion capacity increases. So metal insertion with carbon iodine bond is very easy, and it becomes a little bit difficult with bromine, and it becomes extremely difficult with fluorine, and fluorine never happens.
Any other question from audience? No? If that is the case, then once again, I request all of you to join me. Thank you, Mr. Morgan, for delivering very nice talk.
May I please kindly request Chair to please present the memento do so. Thank you so much, sir. There's a small announcement. All the participants, they are requested to please collect the certificates from Friday evening, that means today to Saturday evening, at the certificate committee desk in the convention center lawn. You also check your poster, oral and attendees number from the board placed at the certificate committee desk. Please kindly check your details before you get those certificates.
So now last lecture of this session is going to be delivered by Dr. Jayadev Laha, which is currently working as a faculty in NIPER Mohali itself in a process chemistry. He is a PhD from National Chemical Laboratory, Pune, basically belongs to West Bengal, and done postdoc from US, from the University of Carolina State University, and Mayo Clinic in United States, followed by he worked as a faculty member in drug discovery in neurodegeneration, Harvard Medicinal School. Dr. Laha produced several PhDs and postdocs in NIPER, and also trained more than 60 master students. Dr. Laha's group is involved in demonstration and translational application of the laboratory concept to prepare a marketing drug. And he has several publications and patents to his repute. He is also referee to ACS, RSC, Science Direct, and Willy journals. So now, on behalf of audience, I would like to invite Dr. Laha in his own institute, to in his own maximum of his audience, to deliver talk on sustainable synthetic process for the preparation of pharmaceuticals beyond academic research.
Hello. Am I audible? Hello.
Yeah, Dr. Laha, please.
Hello. 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 Yeah. Thank you very much, Tiwari ji, to introduce me in my institute. So, I'll give you some background about how I became the speaker today. Actually, I'm coordinating this session, so I'm not supposed to be a speaker. But then, unfortunately, two speakers, finally they turned down their plan to come to NIPER. And then we are looking for 2 additional speakers. Somehow I was able to find 1 speaker, and I'm very much thankful to Murugan that he accepted our invitation with a very short notice. And then again, organizer is asking me whether you can give a talk. And I was telling him throughout this year, I already gave 3 times. And probably some of you will be looking at the same kind of slides and will be accusing me, we have seen the past. But then just to gap this buffer, I have to give this talk. And sir, actually that's the thing, like why your talk was at the beginning. I wanted to say that. So after the lecture given by 2 industry speakers, it is very difficult to convince you what we have been doing because I try to divert you toward the academics research because you are pretty much with the industry now probably. So what I'll try to talk about today, the research which we have been pursuing in the group. Of course, as an academics person, our first priority to be very truly publications. Because the mandate of the academics institution and industry is really different. Because we are in academia, we have to produce human resources, and they need publications for their subsequent growth. Because without publication, they cannot go for higher education or higher professional experience. So if someone is ended up with 1 or 2 publications, these days it's very difficult to get even 1 single postdoc. And these people, when they're now they're sitting in the industry, Murugan has so many publications, or Dr. Tiwari has so many publications, we work together. So I'll try to divert you a little bit to the academic research, but then I'll also show you something we do beyond academic research. It's not very much restricted to only publications. We also try to do something which is beyond academic research. There is a potential, maybe industry may be interested later, if not now. And that's what our goal is. So we'll talk about sustainable synthetic processes, which is actually, this terminology is very much utilized these days. There are various organic reactions we discover in academia, but then maybe they are limited to only publications. But I'm talking about those kind of reactions which industry would be interested in. They may have a look, try to explore it further, and that's how they may find something interesting. But I'm not saying that the academic publications are very much limited to only academic publications, because I know I happen to be a part of a team in industry And we have been assigned to make one compound in maybe kilogram level. And we just followed a TL procedure. And the experimental procedure is very much limited to that particular thing. But we were able to make that compound using that TL procedure. So it is useful. Academic publications are useful, but then of course industry look at the feasibility of the process. And that's what I'll talk about, how we try to find some feasibility which may be interested to be interested in later. Am I audible all the time? Okay, all right. So now right after I said about the academic research, again I'll try to divert you to that point. Students, not only the faculties, students are also very much interested with the publications because it is about their career. There are several students sitting in the auditorium from our group of different department. I know they're very much interested in publication because that's what their future. So one in the next slide itself, I'll show you something interesting slide. Something, this is a template for some RSC journals. But then, these students, they are getting married. They are so much into the publications, their invitation they made in such a way that this is a journal, some manuscript can be submitted to publications. And I got from this one, it is very interesting to see, it is actually, these students actually work with my wife, and wife actually also forwarded to me. And I was so much surprised when I was sitting at the hospital for eye cataract surgery of my mother-in-law. I was so much surprised that students really, they are, motivated for research, certainly, but then also they look for their career. So anyway, so I just tried to divert you to the academic research so that you don't accuse me later compared to what has been described earlier. Oh, sorry. So in this slide, I show you various synthesis of the heterocycles that we have been doing in the group, we have made in the past. And these, as you have seen in the past, heterocycles are really part of pharmaceuticals. And I would say rather over 40% molecules, pharmaceuticals, they comprise of nitrogen heterocycles. So synthesis of nitrogen heterocycles is very important. And it is always my passion that how these heterocycles can be prepared very quickly. Because when I studied at Harvard, many times, the director, the medicinal chemistry director used to come and tell me, can you make this molecule? When I look at the structure of the molecule, yes, I should be able to do this. How long is it going to take? How long time do you want? You cannot talk about months and years. You have to talk about in weeks or in days. So if you go for the literature synthesis that are available, they are many times stepwise, multi-steps. Can you make such kind of heterocycles in one step? Very easily. I'm not even say all the time easily, but can you reduce the number of steps? So with that context, we have developed a couple of various heterocycles we have developed. We try to address some of these aspects which we really mean convergent synthesis, domino and tandem reactions, and transition metal catalyzed oxidative synthesis and all. So like if you go for the domino reactions, it is a very fantastic development. And it is really can be tool for a process chemistry aspects. There are various bond formations. Maybe one is CC, one is CN. But only one set of reagents would be able to do this reaction. one set of reagents, more than one bond formation. It's a fantastic deal, like you see the deal with the pizza also, buy one, get one free. So domino reactions, tandem reactions, they are such kind of reactions, they can be performed, more easily, but then it requires really exercise. One really has to find a particular condition where they can be performed. It's not that easy that you think about something and dump it, you get the product. Because you are talking about various kinds of bond formation under one set of reagents. So such kind of things we have developed, even the double-stage functionalization. So this is a very hot topic these days. Earlier, people used to have two functional groups to have the bond formation between the carbon-carbon. Later, it is displayed just only one functional group, other side CH bond. Now people talk about only CH-CH. And just you dump a cheap reagents that can give you oxidative synthesis. So it is much more facile than what we have learned earlier. So using such kind of techniques, we try to make these heterocycles. But again, I'm saying not all of them will be useful in industry. Don't take it as granted. That is our apprehension that if we could do something industry may be interested in, and then they may have a look. But of course, sustainable synthesis, some of them are of course sustainable synthesis, and that's what I'm going to discuss in the next couple of slides. So, here I show you different kinds of APIs and KSMs that we have been making in the group. Of course, this practice started, I won't say from the very beginning of my research career, sometimes 2015 or so. And we have been really involved in since then how to develop a method for such kind of preparations of KSMs and the APIs. And we always think about developing a chemistry which is sustainable. Again, when I mean the sustainable, I can explain it the other way. Think about the classical old songs by Lata Mangeshkar. Still people sing this. The modern songs, how many of you sing every day? So it means that old songs are really sustainable. They sustain forever. But the modern songs, they are not that way. So one has to, if we develop such kind of chemistry, then probably industry may look forward to our and to our reactions to the accomplishment. Now, I'm not going into the details of all the drugs. What information I've given you here, various kind of drugs with their, various activities, along with their price and values. So some of them are very high value drug, some of them are very low value drug. When we get involved in this synthesis, we look for high value drugs. Although there are some drugs we have been pursuing in the group, one of them is like, paracetamol. And it's how cheap it is. So paracetamol, 1 kilogram price is only 500 rupees around. And an organic chemist to academic person have been asked, can you make paracetamol out of 250 rupees? It's a really challenge for us sitting in academia and develop a method of 1 kilogram of paracetamol in just 250 rupees. Because the industry said, that's what I'll take you. Because we already get for 500 rupees. You make it half, then we'll get from you. So we have to do, sometimes we are involved in such kind of products because some of the actually KSMs are under PLI scheme, which is Atmanirbhar Bharat, product links incentives. But then there are some other drugs also we are involved in, and we try to design synthetic routes looking at the aspects like process chemistry parameters, sustainable synthetic methods. You also calculate before you even jump into the synthesis to look the price, whether it is the projected price is going to be lower than the market price. But then, it is sometimes difficult because when students work onto the project, they have been assigned a particular things. Sometimes untoward results get them more motivated. And that's what sometimes we get distracted. But then yes, we are onto this such kind of track. We try to develop such kind of KSMs and APIs. And what kind of, reagents that we choose for, that I'll just try to highlight you. As I mentioned earlier, when you look for an oxidative reaction, can we apply a very cheap oxidant? And that's what very much, using persulfate, we have developed so many actually reactions. And indeed, we are able to make at least a gram scale. And I'll show you some of these reactions, not only for CC bond formation, also for CN bond formation. Similarly, a reductant. I'll show you why such kind of reagents, because they are very cheap. And they are actually used in industry. Looking at those aspects, we try to gauge this interest, whether if we develop this chemistry, probably industry will be interested. And that's what we will today talk about, some of these aspects related to maybe telescopic process. You already know what is telescopic process. So some of the students, those are taking classes. My personal, I'm pretty sure they're taking classes. So something, without purification in each step. And can you do couple of steps? Can you go for a couple of steps and finally you purify the product? That's how it could save your money, your effort, your time. And that's how telescopic process is one of the important aspects in process chemistry. We are trying to adapt here. Besides telescopic process, I'll talk about other reactions already mentioned, tandem, amidation, and cyclization. So today we'll focus most on this such kind of reaction, like how to do nitro group reduction, and also how to do amidation reactions. And I'll show you some of the applications of these reactions, in synthesis of APIs and KSMs. So the first one I show you, reduction of a nitro group. Students are very much familiar with this reduction. The first thing it should come to your mind, or it usually comes to our mind, reduction of nitro group is very much achieved using a metal and corresponding a hydrochloric acid, or maybe some sorts of Bronsted acid. So looking at the aspects of green chemistry, we started thinking, can we develop an alternative to such kind of nitro group reduction? Because though after the reaction, it produces, and many times if you want to reduce this nitro group, especially if it is present in the nitrogen heterocycles, the processing problem is going to be very difficult. And since you are using the hydrochloric acid, if you have an acid-sensitive functional groups, it is going to be difficult to use such kind of conditions. So we are looking for some more chemoselective, not only avoid any acid or metal, also we want to see whether a chemoselective reagent can be developed, where nitro group can be reduced, as well as if the other reducible groups, like in this case I'll show you aldehyde group. And that's what this method we developed very recently, where we are able to show you here, when you have an aldehyde group, also have a nitro group is selectively reduced to NH2. And then if you have an aldehyde group, that of course it uses synthetic handle for the further in situ cyclization to give you the corresponding such kind of heterocycles. So this is a certain development. Of course, we have shown only one case here. We are exploring more, and we are looking for how this can be really applied, because some of the molecules is already here. So there is a sunitinib drug we are working on. And in this drug, the primary thing that you understood how to prepare the starting material, KSM. And that's what we're looking for. Some of the things I really did not disclose it because for patent filing issue. I did not disclose you, I cannot disclose you how we have prepared this because it is patent filing is in progress. So we were able to make this KSM using nitro reduction, this reagent. And instead of 3 steps actually, now in 1 step we are able to make this KSM. Such a huge development. I mean, we are able to start this cell only just gram scale. We are working with the more, not only 1 drug, we should be able to probably make more drugs using that one. I'll move to the other class of reactions. I did not mention Okay, so coming to I'm not going too many reactions because I also had other risks to show here. So this is something related to, I just show you the entire slide. When we talk about sustainable synthesis, we try to learn something from nature. Because nature is one of the sources to learn organic reactions. And when you look at the reaction, this one, there's how isoneazide actually works. And when you looked at the mechanism, it's very interesting to us because already we have been working on such kind of acyl radicals. And before this also we have developed so many ways, different substrates to form acyl radical. But then when we looked into the literature, we did not find how such kind of pyridine acyl radical behaves. And the radicals, they're actually reactive intermediates. It is very difficult to trap them, but then you have to make it react very instantaneously. Although 10 or 20 years ago, these radical reactions are very much limited to polymerization reactions. Industry did not think about whether such kind of radical reactions can be performed in organic synthesis. But then these days we have been using academics researchers very much involved in these radical reactions. Especially we became interested because when we looked into literature, we did not see much reactivity of this such kind of pyridine acyl radical. Because nitrogen can be moved from 2, 3 different 4 positions. So you could have various pyridine acyl radicals, at least 3. So even those reactivity were not explained in the literature. So when we looked into the mechanism, we found that how does it react with the NAD+. So when this part is the positive part, electrophilic part, so this is then your electron-rich part. And that's what we understood that this is, although pyridine is there, but it's initially we thought it may not be that, it may be rather electrophilic, but it is actually nucleophilic. But there are some cases when I'll show you in the here also, how that can be reactive in a different way also. So we tried to be understanding this one, we used such kind of isoneazide like carbazide as the starting material. The question was how can we prepare in this radical in the laboratory, not under the biological conditions. And that's what such kind of reagents, TBHP, which is very cheap, byproduct is only tertiary butanol, can be utilized to prepare such kind of radicals, unlike nature, the way it is shown. And that can be reacted further with amine. And this word actually, if you really understand, sometimes one could, it is difficult to understand at the beginning how does the reaction happen. So in this case, when I say this nucleophilic, your amine also is nucleophilic. But then we have also shown in other cases, it can be made ampoulant under the condition itself. So one becomes electrophilic, one becomes nucleophilic. And that's how the reaction occurs. I'm not going into the details of all this. So based on this, we were able to make various amides, especially pyridine carboxamides, that are not very much known in the literature. And see, this amide is entirely different way we prepare, the way it has been, the amide is prepared in the, in industrial literature. People take acid, or maybe it's acid amine coupling reagents, or its corresponding, activated derivative of acid, like acid chloride and all. So, we also are planning to prepare, we already prepared nicotinamide, but then, it can be, we are pursuing more about this one. So such kind of, another thing I just want to show you here, the water is also we are using in a reaction. So initially, we become very interested when we learn that industry, in terms of green chemistry, water is very useful, We become very interested. I'll show you another reactions where we really talk about synthesis of pharmaceuticals in water. But then lately, learn from industry people again, well, if you develop reaction in water, how can you do in reactor? So whether its reaction is in water or on water. If it is on water reaction, means after the reaction, your compound will be precipitated in the reactor. So how are you going to transfer the product? So it is really for us also a learning process, talking to industry people, and they sometimes they give us good advice. But then I saw some of the reactions with Morgans and others, that yes, that reaction can be made in water if you add some, let's say, TH-based solvent. But then, we just wanted to show, can you really perform the reaction in water? I'll move to some other aspects of process chemistry that you are building in the group. How much time is left? Huh? Time is there, okay. Anyway, after this only panel discussion is there, right? All right, so this is one thing, and we have been working on since, this is the first one we actually started in the group, sildenafil synthesis. Okay. First, when we looked into the the commercial method, how sildenafil can be prepared, and that's what we learned, that this can be prepared. This is the way. I mean, I'm not going to the details of all these steps because it may take much more time. I'll just request you to focus on these two steps, where the first step is the essentially amide bond formation using CDI. Is a coupling reagent to form amide. And the second was a very strong base to cyclize the ring. What is interesting here, a secondary amide group actually undergoes cyclization with the primary amide group in the presence of a very strong base. And we started thinking how this can be prepared in just one step. Omitting two steps, can you do it one step? Secondly, can we avoid such kind of coupling reagents? And that's what I showed in the previous slide. We are very much interested for amide bond formation. We become more interested since actually discovery of this reaction. So we were able to do this. We are able to prepare this, this particular product, where in one step, two things happen. Amide bond formation followed by cyclization. And two things happen, presence of only one reagent. And that reagent is, as I showed you at the very beginning, it's a very cheap reagent, the potassium persulfate. So, of course, there are certain issues and problems we will talk about, or I'll show you that one. I'll try to project what are the limitations in our method and how try to improve that, such kind of practice. Like here, we are able to show this, and finally we made the drug. But then, as I show you here, if you look at this, we have invariably have to make alpha oxo acid, which actually, when it works under this mechanism, it goes through actually radical reaction. And decarboxylation takes place instantly in the presence of sulfate radical anion. I'm not going into the details of the mechanism today, but it helps with the decarboxylation to form an acyl radical here. That acyl radical reacts with the amine that I mentioned at the earlier. Amide bond formation, and under that condition itself, it undergoes cyclization to form this cyclase compound. You do not need any two set of reagents, not even coupling reagents, not even strong base. In terms of limitations, yes, one has to prepare such kind of materials. Unlike here, you have an acid here. So in the next slide, I'll show you or maybe next two slides, I'll try to show you what was our attempt to improve this. We tried to give more improved method, but then I'll also make a conclusion whether that is still workable or not. So I'll go to the next slide. Here, we tried to show you, sometimes ago we had a lot of discussion with DMSO problem, right? When we first have done this reaction, it's a fantastic reaction. In a sense, you do not need any reagents. It's a reagent-free synthesis. But one has to use DMS as the solvent. And the reaction is at room temperature. Perfect for industry. But I will say you don't go for this method, because there is a processing problem. So when the product is formed, sometimes even it's not even a complete conversion. And when the product is formed, the question is how to isolate the product. That's what we had the discussion with Dr. Swain also. What we wanted to do, we add some non-polar solvent to the DMSO. Try to precipitate the product, because this product is very much polar. So when you reduce the polarity of the solvent, it started precipitating. So we are able to do this, but then really I'm not pretty sure whether it can be done in large scale, because still some product remains in the solution. So such kind of processing problems also we think about what kind of processing problem industry is going to face. We try to work on this. Now I move to the third generation we talk about, where I really try to emphasize that we need to use alpha-oxo acid. And for that, this is prepared actually from acetophenone, using selenium dioxide and all. And when you wanted to file the patent or even wanted to file a paper, publish the manuscript, reviewers were very much opposed to this. Oh, you are using selenium dioxide, it's very toxic. Can you avoid selenium dioxide? And that's what we started thinking, can you really avoid this particular step to make the starting material? We are thinking a different acyl source for such kind of starting substrates. And that's what we got into the problems, how a different kind of acyl source can be utilized. And other problems, I just briefly tell you what are the things. We really thought about what are the problems in our first generation, second generation synthesis. Can you really avoid some of the problems, if not the most? And look at this such kind of reagents, avoid all these reagents. And the first generation, we used potassium persulfate. Reaction was more efficient. But then second generation, it is only DMSO, room temperature. But then reaction was, but isolation difficulties. You will ask me that how did the reaction happen at the room temperature? And that's what DMSO has a magic role to play there. And when we understood the problem, now we started thinking about if we have to omit even potassium persulfate, because we have been using stoichiometric amount. And that's what industry says, even this green chemistry principle also says, avoid using stoichiometric amount. Can you think about catalytic reactions? So any oxidants where it is stoichiometric amount is utilized, the idea is can you make it catalytic? But so far, there is no literature where persulfate mediated catalytic reaction has been developed. It is still at the conceptual stage. We have been thinking how to really implement it. And that's what, in this slide I just show you how at least we obviate one problem, avoid use of any alpha-oxo acid, use a different acyl source, which is readily available. And now the reaction can be performed in water. Of course, we are using stoichiometric amount of persulfate. So, it's a generation changes, something better than the previous. And that's why we try to show you different kind of aspects in terms of improvement from the first to second to third. And, somewhere you just try to show you here cost comparisons. Although it is, at this stage, very trivial, because, these cost calculations are based on actually the sigma Aldrich price. These are not real calculations, okay? So I'll talk about more about later. Of course, we were able to develop a method using a different this source, potassium persulfate. And, still if you ask me, so do you feel it is the last one? Does it require further improvement? Still I'll say yes. Okay, I have to do it first now. So give me 5 more minutes. Maybe I have the privilege to have a chair in the session. Okay, so all right, so I'm not going into we talk about the impurities when he was talking about, Dr. Agrawal was talking about the impurity profiling. We also have to, try to understand what kind of impurities they form. Look at this yield, it's still way behind. So the major challenge, how to improve the yield. That's what, one can avoid the, process would be much, much better. I'll go to the next other slides. This is one method again, very quickly I'll show you the way diclofenac is prepared, okay? First is amidation reaction, and second, these 2 steps, how to really cyclize this. For this cyclization, one needs to have, this method uses actually chlorination there. So in 2 steps, they try to do this, and then this is very costly, expensive. When I looked at the Aldrich, it's very expensive. And finally, with this opened up through hydrolysis, you get the diclofenac product. So what we have done, we are able to do this reaction in just 1 step. No N-chlorination. And I'm not disclosing again the oxide and everything because that's again patent filing process. So I'll give you more picture, more real picture probably later because we need to really explore further in gram scale and all. I'll move much faster for the subsequent slide. The last, I think this is the last about. So this is then another drug. It is very important these days. At least initially it used to be as antihistamine, but then this is for anti-cancer. Is very much in the required and is in clinical trials, as phase 3 clinical trials. So when you look at the synthesis of this clemizole, it has been prepared in 3 steps. And the question is, when we want to apply this, can you use any aspects of process chemistry? Again, for this one, I'm not going to the details. I did not disclose much here, so that please don't take any snapshot of this one because it is again patent filing. I'm just uttering a word to make you understand. This process we have made telescopic. No purification at any of these stages. Do not quote me. This is, this is under patent filing. So that's what we are able to show. This process is, in terms of cost comparison, it's much, much better. I didn't have this one because how did we get involved in this project? That is a story. A biologist actually required, some several grams of clemizole. And when initially the professor is to buy from vendors. And I think 1 gram is to cost, I think, 1.5 lakhs. And started collaborating with us, can you make this one? It's a very easy molecule to synthesize. And we found this. I'm not going to details how we have really prepared to make you more real flavor that we are working on the scale up in this case. Not only gram scale, rather at this stage, 100 gram scale, we have planned to do even kilogram scale. And this was, these are the two pictures here. Again, I think I tried to give you impressions about various activities that we have been doing, what we have achieved in terms of academics activities. There are certain limitations, it's listed here. I think all of you now understand, I've tried to mention every time what are the other issues in terms of cost, everything. Many times academics people very easily say it's very cost effective. Yesterday I saw a poster and it's claiming it's very cost effective process. I said, how did you know this? We compared this and asked, what is the other comparison? It's talking about another literature, academic work. So one is to compare the commercial method, not with the other academic method and show that my method is much better. So those kind of comparisons are really required. But then I'll just close this one by thanking some of these people, those who are collaborators, national and internationals. And we are actually linked with some industries. We have filed several proposals for submissions, but then it was not granted. But then still we're in touch with some of these industries. Finally, I want to acknowledge all these funding agencies, because without funding agencies, and also most importantly, the group, because of that I'm standing here today. Otherwise, my existence I do not exist for, for such kind of activities. Thank you so much for your kind attention. I'd be happy to take any questions.
Thank you, Dr. Laha, for introducing new chemistry in the process.
Thank you so much.
Manufacturing process. So now floor is open for question and answer.
Hello.
Hello.
So, sir, you have, it's a nice talk. I was wondering whether when you are using phenyl acetic acid and you have done an oxidation, so did you observe the phenyl glycolic acid, or it was something else?
It doesn't form phenyl glycolic acid. It actually goes through aldehyde, because it is an oxidative reaction. Reaction is in persulfate and water, so it gives you aldehyde. That's what our mechanistic studies gives. We tried to establish whether it goes through, oxo acid. It doesn't go through in this case.
So that means it says first a decarboxylation happens and then
Benzyl radical is formed that undergoes CH2OH, hydroxymethyl aldehyde, I guess. Any other question? Looks like students are. Yes, yes. Yes.
यहाँ पे आपने क्लेमीजोल जो
Clemizole,
है,
yes.
हाँ जी, किया है. ये एक एलर्जिक ड्रग में भी आता है जैसे, है ना, तो स्किन के रैशेस में, इचेस में,
Yes.
तो ट्रीटमेंट में. तो मेरा ये मानना है कि जैसे आप इसको सिंथेसाइज कर रहे हो ऑन द बेसिस, तो मैं ये पूछ रही थी जस्ट एंड आई एम आस्किंग कि क्या आप इसे कोई सॉफ्टवेयर लेवल जैसे आर्टिफिशियल इंटेलिजेंस का इस्तेमाल करके, कैड का इस्तेमाल करके, क्यूबीडी का इस्तेमाल करके, इस तरह के किसी सॉफ्टवेयर पे भी इसको लाकर आगे सिंथेसाइज कर रही हैं, ऐसा कुछ मतलब लाइक?
Usually we are organic chemists. So far we have been at least academic level, we don't do it. But then yes, there are some sorts of design we always think before that, during designing the synthetic routes. Okay, that time one could get help from any computational studies. One could design this. Even these days there are softwares available with Reaxis. They give the synthetic design. So it's your choice whether you go look at the synthetic design, because when we design maybe a couple of synthetic different routes, and you try to understand the price. At least at this time we are doing that, and if where the price is relatively cheaper than market price, we take that particular synthetic route. Also we look for whether it does not involve any reagents that industry will not like it. So such kind of practice we do, but not really artificial intelligence. No, those things we don't do. It's a very organic chemist.
Okay, thank you, sir.
So any other question from audience? If there is no question, then I call off this session again.
So I request.
चलो फोटो खींचे, फोटो खींचे. He is my former colleague. Thank you so much, guys. So this was the session. Hello. This session is closed. Now we are going to have a panel discussion. Acha, you guys leaving now? So panel discussions essentially it would be helpful if you all are here. It would may not be much longer, maybe half an hour or so, because people from industry, they are here. So if you can have a seat for maybe another half an hour, or if you really have to go, okay, I don't stop you. So that's what, like, if you have any kind of questions that you did not ask before, especially to people from industry, then this is the right opportunity. Probably I also have some questions, I'll ask them. Maybe it would be useful for some of us. Want to start the session? Yes. Yeah, please, I welcome Dr. Ketan Agrawat and Murugan, my former colleague, yes, and then Dr. Tiwari ji. अच्छा हमें नहीं चाहिए. Oh. Yeah. Maybe your session starts at 12:30, right? ठीक है, आप लोग आ रहे हैं. ठीक है, आपका जहाज भी इधर ही लेके आ. Yeah. Hello. Hello. So again, I welcome three of you to this panel discussion. So this is something related to challenges in the synthesis of pharmaceuticals, key challenges, pharmaceuticals and KSMs. It's working, okay, that's good. Okay, so already we have heard all these talks from the speakers. If you miss anything during the after that presentation, if you have any questions related to this one, if you have any question that really is a burning issue for you, or you are trying to understand something, even if you are not involved in that things, it is fine enough. So, because we have the students from medicinal chemistry and process chemistry department, various other departments also. And also we have expert on impurity synthesis. So if you have any such kind of questions, please raise the question. This discussion is very much related to challenges in synthesis of key starting materials, which you say as a KSMs, or pharmaceuticals, like APIs. So we can start having some questions from the audience first, then probably I'll... Okay.
First of all, I'd like to thank Dr. Laha for pointing out the importance of your publications. But from industry point of view, I think Dr. Murugan also will support me that always look for publications with the commercial applicability also. It should not happen that in a rough word, it should not happen that nobody uses your process.
Yes.
So always look for the commercial applicability also with your processes and whatever idea you suggest. So it's academic plus it has to be a commercial applicability also over a period of time. So look at those angles also when you try to generate a new idea and then when you try to generate publish your paper.
Yeah, so as the beginners, for you, my advice would be that, so when you are performing a reaction, you isolate the product. Okay, maybe you report 60%, 70%, or even 90%. But what is more important is, at least as a student, you guys are, look at what else is there in your reaction. Okay, yes, you have got the desired product, isolated the desired product, but what else is there? Because organic chemistry is not like completely understood, or we are able to map all possible reported reaction into one place. Because if you look at my PhD days and all, this CH activation is never heard of. Okay, so when we do the reaction, we don't even count the impurities or anything related to CH activation. Now you see almost all possible transformation on the aromatic ring can be performed with the CH activation. But if you look back, even our days, we had similar reagents being used in different process. What would have happened is we would have just missed it, or looking at the complex stuffs on the bottom of the TLC, will say just cut them off. Okay, I load into the column and just isolate my product, forget the rest of it. But why I'm bringing this up is understanding what kind of impurities are forming can lead to something else. Okay, I myself had an experience of developing a great process just from an impurity from another process. And we had even published editorial letters from those days. Okay, so likewise, so while at your level, I don't think you have that much of pressure, to push the process and go for the manufacture. So you do the reaction, you have a privilege. You can take the mother liquor, you can take the residues, anything. Do, and you guys have, the high-end equipments with you. You can do MS, you can do MS, LCMS, CMS, LCMS, MS, all possible, technique are at your disposal. So you could always analyze those, find something, find for some opportunities, right? And you may, who knows, you may discover something great. Yeah.
I want to add further what he said. So now, since morning you see, and from academy, from industry, concept to commercialization, it's long way. So now where you are, the training position, you are basically training for a chemist. Here you are understanding the concept of the reaction, byproduct is very important. And then with that time, beginning itself, the overall concept to bringing academy, industry together to inculcate this culture. Like when student is getting trained, they should be knowing that what they are doing, can it be commercialized and what kind of order. Right. So when you are learning stage, what Morgan says, we should be knowing what is the way product, what is the side reaction. Many times it leads, bigger innovation than what you are doing. When you know what you are doing, that is not innovation, right? You are executing your idea. You get something and you find out how it has happened, then apply, that is the innovation. In that, during PhD, everybody, we have learned that after reaction, we do brine workup, right? That's the very, means it's a practice, everybody does. When I went to Germany for postdoc, I also did that. Those people were not doing. And during brine workup, what I saw, one impurity, it forms chlorination. Brine has given me chloroproduct, nucleophilic substitution with brine. Then I was thinking how then I just left it for 3-4 hours. My total product converted into chlorine. For that purpose, I published that result in the journal. Only characterization we have to do. So whenever any minor changes, then that's what experimental science, observation, observation, observation. 80% is your observation. Remaining your basic chemistry skills and whatever. Now, again, we want to take this discussion in what is the objective of final discussion we have. Now, Dr. Kirtan has started with nitrosamine, very simple molecule, you see, right? It's a carcinogenic. It was known, I think, around 60 itself. It is a carcinogen, but it was overlooked. Pharmaceutical industry did not bother on this. At that time, that label, I think, instrument was not able to identify it is a carcinogen, and it's a known carcinogen, and you know how it works. This NDMA we call means nitrosamines in body. When it goes to your body in enzyme or some enzyme is there it becomes diazonium and it alkylates your DNA and it mutates. So either this generation or next generation will cause your cancer. That's why it's a very serious issue for pharmaceutical industry. And we are bound to use some nitrating resin or when we are a kind of chemistry, some other chemistry. As a chemist, you can think, can we avoid those chemistry? Right. Being in academia, you have a liberty, you have a time, you can think. Industry, people cannot think. Because we have to deliver, time-bound delivery is there. Because industry is meant for making money, profit, right? Academia, here you are at learning stage. Everybody is learning in that environment. I think this kind of challenge. It is not only for publication, it is for serving better cause, means human, all patients you are serving. Lot of efforts, money is going to solve this issue, right? So that way we have to go. Now Dr. Murugan presented very nice chemistry concept, process chemistry concept. So from there, if you have any question, any doubt, this is the team can clarify more better. Nitrosamine is there. Dr. Laha always is there with you people. Your guy, professor. So this is the objective of final discussion. And if you have any question apart from lecture, if some question or your doubt was not answered, you can now raise those questions.
Good morning, sir. This is Pawan Porwal. Sir, I am working with the concept of some impurity formations in amlodipine, in fact. And when we had surveyed the market formulation containing amlodipine, the sulfonate esters were very high at such site. And in fact, it is genotoxic in nature. We had observed the TTC limits also. Then we were surprised that it is actually at very high power of it actually. And this impurity is coming from the API. Though there is significant difference with respect to the formulation and API, we are observing high side of it, and we are thinking of the excipient might be promoting the formation of those particular one because of the availability of aldehyde, all those things as a part of impurity in the excipient itself. Now my question is that how can we reduce the moisture sensitive impurities in formulation itself? In the last of your discussion, Dr. Murugan, you had, I think so, you had mentioned one API which is actually moisture sensitive impurity was forming in your API. I am just talking about amlodipine bisylate. Bisylate salts is there. What is that? C6H5SO3 is making a ester with methanolic groups, or in that case. So that my concern is that how to reduce those impurities are actually mostly of moisture sensitive, because we are living in a country where the moisture will change from 35% to 100%. Nearly, we are living in Guwahati, where the moisture will be around 90% in 3 to 4 months in that case, sir. And we had observed the same study there also. We had changed the moisture content with respect to RH in that case, and we have observed the formation was escalated like anything, particularly for those impurities. My concern is that I want to reduce the level of those impurities in the formulation. It is moisture, though I am not a chemist by background, I am just analysis, person, so I have a limitation to understand those things. Can you please? Sir, that means the formation of impurity was escalating in the environmental condition when the moisture is high in the. It is bicylates, sir. That is actually methyl benzene sulfonate, that benzene sulfonates, or you can say methyl benzene sulfonates are there. There are some 4 or 5 impurities are there. All are genotoxic. Yes, sir. Yes, yes. Not methylates, in fact, sir. It is bicylates.
There are amines are there which are pharmaceutically acceptable and which will form the salt. Okay, so in that case that doesn't impact the bioavailability. Okay, but only thing, yes, you will be coming with a novel formulation, then to go to the market, then you need to spend significant amount in the clinic to get the bioequivalence done and other thing. Like I basically don't work with the innovator formulation. I mostly work with this either innovator in the sense that we are inventing product, then we know that, say mesylate I had, so we know that you should not have any alcohol related stuff, even including the carbohydrates. So your excipient should be non-carbohydrate because you have methane sulfonic acid. Okay. So likewise, you design, because those people were aware of because this was done after 2005. So from 2005 onwards, people are became aware of this mutagenic impurities, and these alkyl sulfonates are the first of those few identified them, right? So people were aware that okay, now if I have sulfonate or methane sulfonic acid or benzene sulfonic acid, any sulfonic acid, then I should not have alcohol into my process or formulation.
I want to add what Murugan says. Most of the dippings are crystallized from ethanol. Ethanol, methanol, mixture, ethanol, water. In your molecule, it means PA, still ethanol contamination is there because limit are 5,000. They will say, okay, up 2,000, 3,000 ppm, it is meeting my specification. Then during formulation, when you are raising temperature, this impurity may form. So you have to look at your API specification. Ask ethanol should be 10, 20 ppm kind of thing, or 50 less than that. So this thing, many things.
So I will leave chemistry to experts. Have you looked at your facility qualification or something? Is it the humidity in your moisture affecting your formulation process? With humidity control. Okay. 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 But that is the another aspect also you should look in, because there might be a factor that external humidity is affecting your processes. Maybe you can look at that also. Okay.
So you are in academic research. anybody, any manufacturer who is manufacturing API, you can request 10, 50 gram. Okay, I want for research purpose. We will acknowledge. They will give from laboratory. To buy it is a very costly. For them, 50 gram, 100 gram is nothing.
Any other question? From students? Huh? Then they have to charged up.
So it's a rare opportunity. You get various experts from various domains at same place. So you can ask.
Yes.
So my question is very different. Sorry for that. So because I'm working on that, so my question is like that. So sir, my mainly that work which I am doing or I want to do, because I want to do only research. I don't want to go anywhere else. No job, nothing. So according to me, I want to do research and a lot of articles which I have seen on different PubMed, etc. and etc. about artificial intelligence, about CAD, about everything which I am doing. So I'm saying that I'm not getting that point in a lot of articles that if you have a nanoparticles, SLN, NLC, if you are doing that, like nano structured, you are making for drug delivery in the neurodegenerative diseases. So I am saying that if we use the CAD, artificial intelligence, and with the help of that, we made our delivery easy in intravenous in the neurodegenerative diseases to procure that, Alzheimer's diseases, Parkinsonian diseases. So how we can do that in an easy way? It make me to learn easy way by different articles to study that how we use artificial intelligence, different softwares to procure the neurodegenerative diseases and make our delivery easy with the help of nanostructured delivery systems.
Yeah, yes, of course.
We are working with this type of concept in different way, in fact, and whatever was there, artificial intelligence should be applied for the particular meta-analysis type of study. We had diverted this to MPT profiling in that case. You just to define the particular parameter that you want to study, then you can perform some discriminant analysis works, and then you can definitely get it. Means the effect analysis you can do with the discriminant scores, like in SPSS you can do this type of study. It is easy to do it also. Means decide your factors, then you can do it. Means which particular factor study you want to perform. Suppose that I want to understand the formation of impurity at 70% RH. Whether it is forming or not, that is a factor to me. So that I am taking that factor, then result response I am taking second way. You should choose a factor, you should choose a response, then you should design a particular LDA in that model in that case. ANN also you can try. That you should go with the DOE experiments also now. DOE, you should design some in vitro, then you should go with the DOE experiment. That can give you the answer of it. Number of literature is available on that. DOE experiments.
Thank you.
Your question answered?
Yes. Yeah, a little bit, but I'll scroll more for that.
Yeah, so the thing is, artificial intelligence again is a very large term. So basically, when you want to look something out of the software, the feed into software is important.
Ji.
Okay, so what your system is going to do is that it's going to cook up several data that you have fed in and then going to give something.
Ji.
So you also need to give direction to the system. Okay, this is the kind of data that I'm going to feed, and this is the kind of output there I'm going to get. But then your data that you feed is more important.
Ji.
Okay, so again, it is still to your mind or your imagination what kind of data may give me a better solution.
Ji.
Okay, so this is a large world. see the amount of data is available for anything, even if you say what, the permeation thing. We have so much of data available already. But the thing is, what kind of data? Because permeation also is a mechanism dependent. It's not just, log P alone will suffice or something. There are a lot of other things involved in it. Okay, so you need to understand, much better way and look at the suitable data, feed in, then you might get. But this is very complicated.
I understand.
You can go into more depth. Session 7 is just care for pharmacoinformatics and artificial intelligence.
Achha.
Inputs.
You could attend session 7.
Session 7, which will be there in audio. This is the experts of
जी कितने बजे
Any other question? Because we have to finish by 12:30. We have to be there in another place. No, don't worry. I mean, you can have one more question.
Good afternoon, members of the panel. Sir, you had spoken about palladium catalyzed reaction. Sir, can you elaborate a little further on the palladium scavenging processes, how it happens in a larger scale? That's it. That's just my question.
Yeah, so there are palladium scavengers. So the scavengers are basically the modified silica, functionalized silica, or functionalized polymer. They are functionalized with amine or with sulfides. So they are solids. So when you have a solution with a homogeneous palladium, you throw the scavengers, which are functionalized solids, either silica or polymer or whatever the other metal. So your palladium is going to get attached to the function. And stir it for some time and filter it and remove.
Are there any other solids other than silica solids?
There are polymer supported
Okay.
resins are there.
Okay.
Yeah, resins we call. There is a polymer, functionalized polymer solids. And various functionalizations are available. Amine, sulfides, or the thionides, so many other which can bind to the specific metals. And there are scavengers specific to metals also.
So you will add that in a particular ratio or naturally in excess?
It naturally goes in excess.
Okay.
If
Okay.
your idea is it's all heavy metal, you want to get it
Just get
as
rid of
low
it.
as possible.
Okay.
Yeah.
Thank you, sir.
There is a literature. If you go and you see, on PID is also there. There are literature only for palladium scavenger. If you see, there you will find out. Before that, one should think why palladium is coming. It's erosion generally made. From reaction also you can stop it by selecting right solvent, stirring pattern, and reducing that time. If you take the palladium black, it's the bigger particle size, that erosion will be less. Those concepts you can use. Generally in process we think that.
Any other? So I have one question. I mean, and usually I always think that before I ask the question, let me tell you about the background. So the sildenafil synthesis that I showed you, we use a KSM, KSM, which we buy from Aldrich, which is a plurazole derivative with a amino group, amide group. And we start from that one, and then in one step, we are able to make the sildenafil. Only just we made 2 steps to 1 step using more cheap reagents and all. When we talk to industry people, like 3 to 4 industry, they became interested. Then they started talking about tell us the cost of this one, but it was a premature because we didn't. It was only just 1 gram scale and all. Then they started talking later. Can you first make the KSM? Because we have the problem of making the KSM itself. Because they do a nitration step where they uses concentrated nitric acid and mixture, sulfuric acid mixture. And it is a pyrazole, so it is very lousy stuff. It is very difficult to fish out the entire product during the workup and all. So such kind of problems, like That I understood from the industry. Okay, there is a problem there they talk about. So where can we get such kind of information? These are the problems. Is there any public forum? I'm pretty sure there's not going to be a public forum. Where can you get all such kind of information? Because we are looking for some sorts of problems that industry have been facing. Can we address some of the problems? And in that context, we are trying to develop some alternatives to what industry has been doing. But the specific problem really industry has been facing. Some industry came and people came and asked us, can you make chiral amine reducing amine? Although it could be done, but then it involves the use of the chiral catalyst. I was not that expert, so I said, okay, we can think about, but then I'm thinking about other problems, what I said, such kind of problems which industry has been facing, but then we do not know even these kind of problems. Unless we attend any talk given by industry really emphasize on that particular problem, then you get to know. Like today's, we got to know some of several problems. But then, really the area of what I've been working on, we have been working on, if it is relevant to that drug or that step, we would be more interested to take the problem, rather than if somebody is working on, let's say peptides, because we do not have expertise on that. Maybe peptides guy has to look into that problem. So is there any public forum or some sorts of media where you can have access to these kind of problems? No.
I would say no. But see, again, what I have presented is a product-specific problem. Okay. Now, if you want to take that to academic, then I lose my job. Okay.
Don't worry, we are not that expert.
Jokes apart. What happens is that we work closely with the manufacturer.
Mm-hmm.
So we understand what are the manufacturing difficulties. Again, it's a product specific. I'm not looking at a generic problem.
Yes.
For this specific product, what are the issues?
Mm-hmm.
So unless I work closely with the manufacturer, I may not be able to solve all their problems. For example, I said, we did so much of studies, but what finally was required was, instead of applying vacuum, you need to apply pressure, that's all. But this understanding comes only when you work closely with the manufacturer, which you will not have that. So what happens that your contact is going to be an R&D guy. Now R&D guy, once he gives you some problem, he will think that you will resolve the problem, he will even forget. It's not even in his portfolio, he will do something else.
Okay.
So you may not get all the information or all the challenges that is relevant for your development. That's okay, that is again product specific. Now looking at the larger forum, is there a forum? There is not one. But some time ago there was one, like synthetic challenges. Some people used to pose the problem with the price tag onto it if you resolve so problem.
Okay.
I think it's still there.
Okay.
That is one forum I know that people do public. So what happens is there also, I'm not going to put my real structure.
I know. I mean, that's why industry does not want to disclose
I need
this.
money, right? Whatever I do, I need to make money. So I can't disclose everything to you.
Yes.
Right? So that's another challenge. But if you want to work in a larger, any recent surprise for me that we embarked on developing peptides.
Mm-hmm.
Okay. Now, for the peptide synthesis, I was looking at the monomers. The cost of monomers, they're too high. It is just a protected amino acid.
Yes.
The cost, commercial cost I'm talking about, several $1,000s per kg. So, I can seriously advise you people look into the different semaglutide, liraglutide, all these glutides. Okay, they all require all these amino acid, and they are all published in patents and everything there. If you can, and there are no suppliers in India to supply those amino acid fragments. There in the world also there are only 2 suppliers, and they have monopolized so much that whatever the price they ask for, you have to give. And literally, it is an protected amino acid.
Yeah.
Okay, so maybe it requires a specific processing or whatever. We will not know what their processes are, but the raw materials aren't cheap. Why should they be so expensive? You can very well file your patent. You develop a
Yes.
synthetic process, file a patent for all those commercial monomers, and definitely because at least in Indian chemical industry now they look at peptides as their future. Every big company they are working on all this peptide, liraglutide, teluglutide, you name any peptide, and people are working. And there are at least 20 different peptide products in the market. Okay, so this can be a good idea for you guys.
Yes,
Really
thank
look
you.
into, because I see lot of posters on peptide, my pharmacy.
Yes, yes.
People are working on
So, like, if you see in a bigger perspective and national perspective, academia is meant for basic research, industry for applied. Academia also, and now, so far, both are working apart. They don't talk each other properly. They are training the way they were training. Then they go industry, we see very raw, again we have to train for a year. So these are the gaps are there. Now at least there are forum, people started talking. Like academia, the supply chain for industry as a human resource. Like KSME, so that kind of training. Now here comes what challenges industry faces. Like what Morgan says, these are the basic things which we are dependent on somebody. And you know that one thing, most of I think people are not knowing, chemical industry of any country is the main driving force of the developing the country. Chemical industry, if it earns $1, and other place means it means multiplied by 4. For people earn $4 from your industry. So it has everywhere application. Whatever today you are using, then now most of the things are manufactured or prepared using your chemistry skill, chemistry knowledge. So now we have to grow our chemical industry. Pharmaceutical is a part of one. Agro is there, consumer is there, many things are there. So both has to work together, understand each other's requirement, then only it is possible. And like basic research industry, we don't have time to do new research. That is commercial. While working is something coming new, okay, we are protecting it because we have commercial. And any investor will not wait. I mean, they will not, they don't have patience to wait for 5-6 years to develop a new thing. Right? So that is the basic understanding, and I think we understand each other's role and requirement properly. This is going to be first collaboration for whole chemical society.
If you can look at the recent reports from Indian governments with regard to China imports.
Yes,
so I
yes.
think probably you can take up work on those KSMs or
Yeah,
intermediates,
you
which
talked
are
about that
things
one.
we are 100% dependent on them. Maybe if there is a China factor, but if we can replace those with 20-30% also, that will be a huge benefit for the India at least. So you can look at those parameters, those factors.
So, and like if you see every year, there will be a journal where FDA approved drugs are there. You can find out some of the case, some of the chemistry you can contribute. Because when it becomes generic, we will be using. And if the knowledge is available, any manufacturer will be ready to take from you. And they can optimize and use it. So from there you can take this.
Thank you. Let's close this. Yeah, thank you so much. Let's close this session because we have to attend another session there, especially one of them is going to be judged there. So thank you so much. Let's.
Thank you so much. I think we all have gathered a lot of knowledge, right? So the panel discussion ends. Session 6 is actually ends now. We'll be moving towards, after lunch, session 7th and 8th, fine. Session 7th will be in the auditorium and session 8th is going to be here, fine. So in that case, we finish it right now, but we sir, please. May I request, sir? It's the bouquet for sir. That is really great. Thank you so much. Please present this. Co-chair. Yeah. Thank you so much. Thank you.
So hope it was useful for all of you.
Yes.
Fine.
Hale, Hale, Chick.
Take this slide.
I just need to introduce first.
I'll set it up after you. Okay. Let me connect to my charger also, just in case. ऊपर होगा ना? अच्छा. HDMI आपका कहाँ पर है? This one? Yeah. Okay. That's it, right? It's set up? Yeah, sure. मैं वहाँ बैठ जाती हूँ, then
I'll just start and then the session. Hello. Yeah. Okay. A very good afternoon to everyone present here on behalf of NIPER Mohali. We are going to start with the session 8, which is on medical devices, drug devices interactions path ahead. So I request you to all please kindly switch off your mobile phones or put them on the silent mode so that it is convenient. The chairperson for this session will be Dr. I.P. Singh, sir. And the co-chair will be handled by Dr. Deepika Bansal. I'll just introduce Dr. I.P. Singh. Dr. I.P. Singh is a professor in NIPER SS Nagar in the Department of Natural Products. He is involved into teaching postgraduate and doctoral students of various chromatographic techniques and spectroscopic techniques. He's done his MSc and BSc and MSc from the Punjabi University of Patiala, and he has done his PhD in the organic chemistry from PAU. There are 106 research papers, 14 review articles, 10 book chapters. He has guided more than 70 students in MS and 16 students in PhD. He's collaborated with various universities like National Center for Cell Science, Pune, National AIDS Research Institute, Pune, and then University of Mississippi, USA, and various other universities. So he has a wide experience And now I'll introduce the co-chair, Dr. Deepika Bansal. Dr. Deepika Bansal is an associate professor in Department of Clinical Research at NIPER SAS Nagar. And she has received her MD and DM in clinical pharmacology from PGI, Chandigarh, and also remained as senior research associate there. Today, she'll be present with us as co-chair for this particular event. So I would kindly request the chairperson and the co-chair to please take their respective seats. So I would request you to please open the session 8. Technical session 8.
Good afternoon, everyone. So welcome back after the lunch session. So we'll start the session on medical devices now. So medical devices is an important area, emerging area. So in this session, we have 3 lectures from Dr. Neetu Singh, Dr. Abhijit Majumdar, and Dr. Sanjeev Kumar Gupta. So our first speaker of the session is Dr. Neetu Singh. So it's my pleasure to introduce Dr. Neetu Singh. She obtained her bachelor's and master's degree from University of Mumbai and a doctoral degree from Georgia Institute of Technology, Atlanta, USA. She obtained her postdoctoral training at Harvard MIT Division of Health Sciences and Technology, USA. She returned to India and joined IIT New Delhi as an assistant professor at the Center for Biomedical Engineering in 2014. So she has numerous contributions and awards to her credit. She is recipient of Innovative Young Biotechnologist Award 2013 by Department of Biotechnology. She has received Veena Arora Young Researcher Award by IIT Delhi, recognizing best research contributions at IIT Delhi. She is the recipient of Nasi Scopus Young Scientist Award for Women in Science 2019 and DBT's Janki Amal Young Scientist 2021 and SERP India Power Fellow 2021. She is also a mentor for IIT D startup Kriya Labs. She is on editorial advisory board of Journal Bioconjugate Chemistry of the American Chemical Society. So with this brief introduction, I welcome and invite Dr. Neetu Singh to present her lecture.
So thank you so much for that introduction. And let me see if the laser is working. I don't think so. Can I use this one? Yeah? Yeah, this is better. All right, so the title of my talk today, I've given it as Rethinking Solutions for Healthcare Technologies. And what I'm going to tell you in today's talk is basically some of the ideas that we're using in our lab to develop technologies for future. So none of it is there in the market right now, but we are hoping that within maybe 20 years or very optimistically maybe 10 years, we'll be able to get few things in the market. So why rethinking? Yeah, I won't be able to change the slides though, so okay. Yeah, sure.
इसको ऊपर की तरफ कर लो, near your mouth. Yeah.
Okay.
ऊपर से, just a moment.
Okay, so what I'll do is, why am I calling it rethinking? So the reason I've chosen this word rethinking is because I would like to emphasize and I would like to convince you by the end of the talk is that the same existing materials that are there, we are just trying to come up with a slightly revised version of the technologies, a slightly different approach towards developing these technologies, which is going to help us get better diagnostic or therapeutic technologies. And that is the reason why we've called it rethinking. Just to give you a little bit idea of my training, as you heard from my introduction, I'm a trained chemist. So all the aspects that I'm going to show you today will have a little bit of chemistry aspect to it. And I would like to again highlight how using that chemistry, we have been able to rethink these solutions. So, you know, all of you are aware that the two big problems that are facing in the world, you know, in healthcare is basically if we can detect diseases early on or come up with better prognosis, or in case we're not able to do that and there is an organ failure, we can come up with strategies which can help us regenerate those organs or even completely create an artificial organ. And these two are the broad areas that my group is also working at IIT Delhi. So we are basically working in areas broadly in disease monitoring, where we would like to come up with technologies which can allow us to monitor diseases, do fundamental studies so that we can understand what is happening in the host when these cells at a cellular level, what is happening in these diseases, and we can come up with some insight which can be used to develop better therapies. So that's the goal for our work in disease monitoring. In the second part, we are working on engineering organs. As I mentioned, it will be nice to engineer organs. Again, we are looking at fundamental understanding of cell-cell interactions and cell ECM interactions and trying to see if we can manipulate those interactions to regenerate organs or to create better organs. We are working on, so I'll give you a glimpse of these two things in a little bit in the next few slides, but I'll get a little deeper into the new drug delivery vehicles that we're working on in the lab and why there is a need to develop better vehicles. So for disease monitoring, just quickly telling you, when we started our group in 2014, we asked a very simple question. Tumor biopsies, we all are aware, is very common and it is very easily available. Can we take these tumor biopsies, let's say from patients, and so primary cells, being ambitious, and can we create multiple such tumors, right? Multiple such spheroids or tissues which has these tumor cells, right? Can we basically break these tumors, get the cells, which is a very common thing, and then take these cells that we have and bring them back into a aggregated cell or a tumoroid, if you want to call it, right? What will that allow us to do? Simple, from one patient, if I can create, so that is a precious biopsy, right? It's a very precious sample that comes from the patient. So if I'm able to create, say, 400 or maybe 4,000 and whatever numbers you want to do, if I'm able to create so many such tumors from the same patient, now I can do all the drug screening, which can be for that particular patient. So it can be towards personalized medicine. I can also use them to understand whether the tumor samples are aggressive or not, whether they can metastasize faster. So I can come up with platforms which can help us study those things. They can be used for fundamental studies, of course. So I can address all these questions that I've listed here in solving the problem of treating cancer, right? So that was the goal. And these are some of the work that we've published and some patents that we have from our lab. Those who are interested can look into these articles. Basically, what we ended up doing is so the same material, collagen, alginate, gelatin, polyethylene glycol, the same materials that you all have heard which have been used to create these 3D scaffolds, we have used the same thing. The only difference now in our system and the system that is used in the clinic, so in the clinic or in the lab today, the status is that if you have to grow these organoids, you get Matrigel, which is very commonly available to create 3D scaffolds. And these 3D scaffolds can give you tumoroids in about 21 days. So what do I mean by tumoroids? The cells will be, behaving similar. All the protein upregulation things that we are looking at, certain markers, will be similar to what it is in the patient sample. And also, the spheroid that we get, the tumor that we get, is going to be similar. It will have the same hypoxic regions in the core, and the shell will have, more looser ECM and things like that. So the architecture is also very similar to the tumor that you see in the host, okay? Not the same, but similar. In our thing, what we did was, we said instead of taking it to a millimeter size, which is typically what you would get, can we create micron tumors? Can we create smaller tumors? So we developed this microfluidic based system where we not only made the scaffold smaller, changed them into microns, but also added some nanosensors which will give you the real-time information of how cells are growing. So basically we embedded some nanosensors. So this is the picture of it. You have a scaffold, 3D scaffold, which is micron size. Typically this is what they look like. And then you can encapsulate your cells, which also has the nanosensors. These nanosensors can detect change in pH, which will tell you about the growth rates. It can detect the change in MMPs, which are regulated and things like that. So we can now tune it to have any readout of interest, okay? With that, now what we're doing is, in the lab, we're trying to use them for drug screening, for organoids, biomarker detection, cancer metastasis, and so on. So as I mentioned, we've also extended this because these nanosensors, fluorescence can be one readout, cyclic voltammetry or electrochemical readouts can be another readout. So what we did was we basically tried this to study even bacterial cells, because now instead of cancer you can put your bacterial cells and it'll tell you cancer cells takes time to divide. So any changes will take at least 48 hours to 72 hours or longer to see the changes. However, bacterial cells are faster. So we can actually detect changes in bacterial cells even faster. We thought we can use it for diagnosis of bacteria. And in collaboration with ILBS at Delhi, we are using this as a platform to look at sepsis patients. With AIMS, what we're trying to do is develop these platforms to detect, to study metastasis. So now we have this platform where our thing can be used, and these clusters that you see, more the clusters, more migratory potential of the cells are. What it means is that the samples that you put in, the cells that you have will have more migratory potential. That means they probably have more metastatic potential. And we have correlated with a lot of EMT markers which are known clinically. So that's a platform we have developed. We have genetically modified the cells also to have less of those mesenchymal characters and try to see if it actually correlates, and we are able to study those changes. And this is in collaboration with AIMS, Delhi. With the company BFlow in Spain, we are now using the same platform to grow liver models on a chip. And we are using this modular concept where we have micron-sized different cells in close proximity to get the cell-cell signaling. So these are some of the things that you will see in future from our lab. Moving to, so that was a simple tissue, which is a tumor tissue, which doesn't have any function, if you ask a chemist. But if you look at the functionality or an organ, development of an organ, it's a long way. So we're also working on developing engineering organs, as I said. And again, this is some of the work that we've published in this area, and you can look up for more details. What I wanted to highlight here is that if you look at an organ, the problem is that in a close proximity of few microns, I would call it, you have two different cell types. Okay, and that's being very simple. A bone is a very simple organ compared to a liver or any other organ, right? So if you look at this osteochondral region, it has this region which has an ECM, which is very, very rigid, which is your bone ECM, versus an ECM which is very soft, or a cartilage ECM, right? How do you create that kind of a system in a lab? It's impossible. The 3D printing techniques that we talk about will not allow you to get this defined region in a micron range. The way it is done is you print one ECM and then you put another ECM on top of it, and you have the cells encapsulated later. That won't work because remember, if you want to use stem cells, you won't be able to print those because the bioprinting cannot happen with these technologies, right? If you take one scaffold and you use stem cells, you know that people use BMPs, for example, for bone regeneration. BMP is bone morphogenic protein, right? So it changes, it allows the stem cells to differentiate into a bone lineage. All the cells will become bone, right? Whatever you do. So you won't be able to control this. So the way nature does it is, it does not use an exogenous factor from day one. It starts to use the exogenous factors or the BMPs or the growth factors later, right, after certain time. So there is a temporal control of when these factors are coming in play during the biological process. The way we do it is we add them at time 0 and just let the cells become bone cells or cartilage cells or whatever. that won't work for regeneration. So we thought, can we control this differentiation by controlling the chemistry of the ECM and allowing the ECM direct the differentiation. And we've done that, and again, this is basically what we were able to do. We were able to 3D print. So this is a scaffold which has 2 different chemistries. You put your stem cells in them and let them grow without any exogenous factors. After 14 days, they differentiate into 2 different lineages, which is similar to what I showed you earlier. We have done these experiments, and we have now moved on to trying to understand how these topographies or how these chemistries changes the differentiation potential. Okay, so coming to the topic of today's discussion, I'll be very quick in doing this due to time. I want to focus on what kind of technologies we are developing in drug delivery. And I'm sure this is, here I don't have to tell you the importance of drug delivery, why drug delivery is important, because all our organic chemist friends are making all these wonderful hydrophobic molecules which are not seeing the light of clinical translation due to lack of delivery vehicles, because they will be cleared out from the system because of their hydrophobicity. So we need delivery vehicles to take these compounds to the site, right, or to even put them into circulation. And of course, a lot of people have worked on this area for decades, and there are journals dedicated to drug delivery. And some of the solutions which are seen as a promising solution, of course, is liposomes. It's the gold standard that we have right now. It's in the clinic. Everyone is using it as a standard platform. With COVID, we saw its use heavily in vaccines as well. And the nanoparticle systems is also one of the new strategies. People have started using it. So there are many strategies. And again, this is the same thing. Why do you want to use the liposomes and the the nanoparticle? Is simple, because then you don't want to use the targeting moiety. Just by increasing the circulation in the system, you will allow them to go to the site by the EPR effect. Or you can also take advantage of the protein corona formation. It is actually sometimes an advantageous system and sometimes a very bad system for circulation. It is a bad process. So you want to resist protein adsorption if you want to increase circulation. Anyways, these are some of the things that we keep in consideration when we're talking about nanoparticles. What is there? What is the status now? We have liposomes. You all are aware. Doxil is a beautiful example of how liposomes have shown it can do better in the clinic. And Abraxane, which is an albumin-bound paclitaxel, which is the concept is that you resist protein adsorption, increase the circulation time by adding albumin to it, which is the most abundant protein which is in circulation, right? So these two are some of the standard things. Now I want you to notice the structure of liposomes. You all are aware already. It is nothing but a lipid bilayer, and we add something called as the polyethylene glycol, the PEG, which gives the stealth property. What is stealth? It doesn't get recognized by the macrophages. There is no protein adsorption. It doesn't get cleared, right? So it starts to circulate slightly longer. So we put polyethylene glycol in the system, and so this is basically what your liposomes are. It's a very biocompatible material. In fact, the lipids are similar to most of the lipids it's found in our body itself, right? So it's a biocompatible material. It has hydrophobic and hydrophilic regions, so any kind of drug can be encapsulated in that very easily. Yes, there are issues. For example, the stability of this, you can't load more drug because it's a self-assembled structure, so the self-assembly will disrupt. So the stability is an issue. It's not very long circulating. It's good, but it's not there yet. Moreover, the incorporation of polyethylene glycol gives you something called as the Karpa effect, or the pseudo allergies of PEG. Okay, it also has, people have also shown that there are antibodies to PEG which are formed. So it causes that allergic reaction to these materials. But when you're treating a cancer patient, these kind of things can be compromised. So it's a solution, yes, but not complete solution. So we thought, can we come up with something better than liposomes? As I mentioned, very difficult to come up with something better than liposomes. Abraxane was another strategy. So you have to come up with totally different strategy, right? You can't come up with a new material. And also remember, if you take the new material route, the regulatory approvals are a long process. You don't want to go there. So it's better to stay with the existing FDA approved materials, right? So we thought, let's look at how nature does it, because most of the time the motivations, nature knows it all. It has all the solutions. So if we can mimic something from there. What we found was that if you take our RBCs are basically nature's drug delivery vehicle. They deliver oxygen everywhere, and they're long circulating. They're the only cells which are in circulation. And there's a beautiful, if you look at the system from a chemist perspective, all I see is a lipid bag which has no nucleus. Wonderful. I don't have to do any biology to take it out. And it has sort of heme in it, because heme is the one which is giving you the capability to carry oxygen, right? So it's something which can bind oxygen. It also has this beautiful biconcave shape, which gives a nice surface area for maybe longer, for the fluid dynamics, of course, but also to carry more of oxygen, right? Because any other structure would not allow that optimum level of oxygen. So beautifully designed system by nature, which is RBCs. So we said, well, if this is just a lipid membrane and has some mRNA and some other proteins and other things in it, can we just take it out and put our drugs in them, right? Can we use it as a liposome? We started looking at what is there in the literature. We found a lot of paper where people have used this. So it turns out that there's something called as CD47 on the surface of the RBCs. You all know it more than a chemist. So on the surface of these cells, RBCs, we have something called as CD47. And this is responsible for the stealth property of the RBCs. What it does is, it actually interacts with the macrophages, has these other receptors which can recognize these CD47. It interacts with them. There is a clustering effect. So they all cluster. And because of this clustering effect, there's a downstream signaling which tells that this is self, don't eat this, right? So it's a simple process. Now think about it. If I put the CD47, so many scientists have recognized this, and they said, well, we don't want RBCs with the liposomes. Let's put CD47 on the surface, right? Just decorate the nanoparticle with CD47. Now think about it. If you have this surface and I randomly put CD47, will my interaction be the same as it happens in this natural process? No, because I don't know the density of CD47. I don't know how close they should be. I won't be able to control all that, right? I'll have to do a random process. I'll optimize it. I'll say this is working better than this, but I won't get to the right solution. So adding CD47 on the surface clearly was not the right thing, and that is why probably it has not given very encouraging results in the literature. Good for a publication, but maybe not for clinical translation, right? So we thought, then the second strategy was that people have used is putting the entire RBC membrane on the surface. Forget about the density, we'll just put the whole thing. That also did not work because that makes the size really large. Larger the size, the macrophages is going to clear it. It doesn't matter whether you put CD47 or not. They are tuned because they just phagocytose, right? So anything large, they're able to clear it up by phagocytosis. So that won't help. So what you want is the RBC membrane, but in nano sizes probably. Again, as I showed, these are some of the strategies which is showing that there is improvement but not there yet. So this is some body of work that we have done, and what we thought was that we will use this erythrocytes, which is similar to liposomes, with their CD47s and other protein surface markers intact. We're not going to touch that, but remove all these other things that we don't want. Take it out, okay? Biologists have optimize that process for us. So it's nothing new that I'm doing. Again, remember, it's rethinking. So all I'm doing is just using it with a different approach. So I have used this strategy that the biologists have optimized of using osmosis to break it and make host RBC particles. And then what I did was, now when you removed all the mRNA, you have just the lipid. This lipid is similar to the lipid that we use for liposome synthesis. If I put it in the extruder will have no clue whether it's the synthetic or the RBCs. I just put a mechanical pressure in it, make them smaller. I become a little ambitious. While I'm making them smaller, I put my synthetic lipids as well. Now I can put whatever I want. because I'm just trying to self-assemble this whole thing. So what I've created is a semi-synthetic system which has these natural vesicles, natural receptors, but also my whatever choice of my lipids are. Okay, I can put in whatever lipids I want. I can put in targeting ligands, anything, right? So that's kind of what we did. And with that, I'm going to skip the choice of my drug and the dye. The choice is based on being able to locate where it is in the liposome exactly. So I'm a chemist, so I like spectra. So any molecule which has unique spectra, it will be easy for me to identify where it is. So these are molecules which will tell you whether they are in the hydrophobic, where they are partitioning, whether they are in the hydrophobic region or the hydrophilic region. So I'll be able to understand what is happening with the system. And camptothecin is a drug which is in clinical, one of the low potent version is in clinical trials right now. The low potent potency because that's the one which is slightly soluble. This is more potent but less soluble, so we thought this would be the ideal system to use for drug delivery. So just giving you a glimpse of the data a little bit, we were able to show that we were able to make sizes of about 180, the entire diameter about ranges from 80 to 200 nanometers, 100 to 200 nanometers. The proteins were all intact. We were interested in CD47, so we did single particle tracking and confirmed with antibodies of CD47 that they were present there. And they were also resisting proteins and other things. When we did some biocompatibility studies, because they're coming from natural sources, we looked at, of course, the basic biocompatibility, sorry, the cytotoxicity and all that. We also did some immune compatibility where we looked at the expression of TNF alpha and IL-6. They were slightly higher, so we did see some reactions at the early time points, but not completely bizarre. Okay, so if you look at the early time points, we did have slightly higher expression of these cytokines. We controlled the drug loading. We then started moving on, looking at them as a drug release vehicles, comparing it to liposomes. I'm going to skip all of this basic data. This is something important. When we looked at the biodistribution, I wanted to highlight how you see a slightly higher amount in the brain, which is very interesting. We don't know why it happens, and we are intrigued by it. The presence in the liver and the spleen is not surprising because it is RBCs, and that's where they get cleared. So we were not surprised by that, but the brain was very interesting. And when we compared it to liposomes, we found that indeed they were slightly longer circulating than the liposomes, and they were present in the tumor slightly more, but they were quite comparable to liposomes. Then we started using them for delivering camptothecin drug, and I'm going to skip this and I'm going to go to this. And we saw that we were able to have a less amount of tumor, so the load reduced. However, we were not able to cure it. Before we cured, the mice died. Okay, so if you look at this data, we were able to not give any survival benefit to the mice. And what we found here is that again, the tumor is not completely cured. Even after, I think, 30 days is what we did, and after that there was a lockdown, so we were not able to. But anyways, if I look at the toxicity evaluation of our thing, you see that most of the places it was not toxic. However, we see a large amount of toxicity, which was similar to the bear drug, even with this in intestine. Okay, so it's not a complete solution yet. So we thought that visit the delivery vehicle or the drug. It could be the drug also. So we wanted to reduce the concentration of the drug and use this delivery vehicle as a co-delivery for something else. So that we can get benefit of dual delivery, right? That's what we did. We used photodynamic therapy and photothermal therapy to be specific, and combined it with the drug, because now that we have the system, we can utilize it to do multiple things. And again, the strategy was simple. When we are extruding, remember I told you the choice of things. I can put drugs, I can put my dyes, I can put dyes which are reactive to light and which can generate heat, like the ICG and things like that, standard things. And I'm going to go to this data. So this is half of the concentration of the drug that we used earlier. So we did not see toxicity, but our tumor burden was also not completely gone. We still saw tumor growing. We think it is the dosage issue now. So now we are addressing the dosage issue to come up with a complete solution. But at least for the proof of concept, the platform works and that can be used for multiple strategies. So you can see that there is a huge jump here at day 14 or 4 or day 8 after a week. So that's where probably we should have dosed it better. But again, these are things you learn as you do your experiments. So not a cure yet, but yes, the platform was working well. With that, we are now moving forward, and there is a lot of things to do in this. We've just started using these platforms, and we've just proved that they are similar to liposomes. There's a lot of studies we need to first compare. We've compared it to liposomes, but we need to compare to other systems also. We need to look at the efficacy of these systems. We need to look at the therapeutic efficacy, the systemic toxicity, what are the survival benefits with this system, and what kind of animals. For example, we've done it in small animals. Now we would like to go to larger animals also. So there's a long way to take this whole technique that we're talking about to the next level. So with that, I would like to end my talk here. And I'm really thankful to my host, my institute, IIT Delhi, for giving us all these opportunities. The IYBA scheme at DBT started funding my lab in the initial days when I had no funding. DST Nanomission, and DBT Nanobiotech has funded this particular project, the RBC project, so I'm really thankful to them. Recently, I got my SERB Power Fellowship, which gives me flexibility in using the funding and gives me visibility as well. The Janki Amal Bioscientist funding, which is for the diagnostic platforms that we are talking about. So I'm thankful to them. And I'm just presenting what my lab does. I'm just I would call myself a manager maybe, an idea giver and all that, but I'm not doing the bench work. The entire work is from this hardworking group of students, which are very, very motivated. I just go to them, give them a random idea, and I ask them, why don't you try this, which has no precedence or which has no literature. They sometimes think I'm crazy, but they end up showing things, and it looks very simple initially, but the story is building up to be a beautiful one. So I'm really thankful to them for making me realize my dreams. Actually, these are all things that we envision as a scientist, right? So yeah, so really thankful to all these students, and we are working in a lot of constrained environments as well, and they've never complained. And thank you so much. I would like to thank Dr. Sharma for giving me this opportunity so that I can talk about my work in this really learned audience. NIPER is a dream for all the people working in these kind of areas. So thank you so much for giving me this opportunity and listening to me patiently. So I'm open to questions.
Thank you very much for the nice talk. The session is open for discussion and questions.
Yes, sir.
Thank you, Dr. Neetu. Actually, I came to know about you from Savneet Kaur at ILBS.
Yeah.
Because I think one of your
That
projects which we
we're
are doing,
yes.
she connected with me for some support.
Okay.
So nice to see you today here at the same
Yeah,
platform where I'll be speaking. So quite a variety of work. I only want to understand that couple of things which you've showed in the initial part. Have you also drawn a regulatory strategy for your products?
I think that's a wonderful question, and that's where scientists like me, we fail. We can, dream things, but we don't know how to make it, how to bring it to the market. So I'll need help, all the help that I can get, and I have absolutely no clue how to go there. I'm just looking like a kid and trying to figure out, looks exciting, but I don't know how to go there. So I'm going to make mistakes. So I think I'll need your help in...
No problems. I think that's something which you should do.
Yes, absolutely. And we are very keen on the platform technologies that we talked about. We are very keen on doing that. So one thing I've done is protected them. So that I'm happy about. So we have US patents, Indian patents. So I wanted to go international as well. So we have protected them with US patents and European patents. Indian patents, we have plenty. I've not shown them there because honestly, Indian patents, I'm not sure what the value is when it comes to the international market. So we would, and most of the pharma companies who are in, or the diagnostic companies who are ready to help with R&D at this stage where we are, is international companies. And it becomes very difficult to go there with an Indian patent. It helps, but slightly difficult. So we would like to have an edge. So yes, we've started that journey, but I don't know how to go about it. So we'll have a talk.
Sure.
Yeah, outstanding talk. Okay, I
Thank
learned
you.
a lot. Okay.
Thank you.
Could you kindly explain or elaborate a little bit about your platform on detecting bacteria and AST? I think this is an outstanding device or the system that we could always use.
Absolutely. So it's a very simple system. As I said, we wanted to keep it simple, and it's rethinking, right? So what happens is when you take the 3D scaffolds are known, people are using 3D to grow cells. It's a very common thing, right? One of the problem these 3D systems are not able to give you information is two things. One, real-time information is missed. So what happens is you do endpoint assays. You kill the cells, you look at what has happened, what biomarkers are present. There is no real-time monitoring of the sample. A lot of things are not known because there are transient changes that happen. You look at the global picture, that what happens after 48 hours. But a lot of small things would have happened over the period of time, which we are missing. So I wanted to detect those changes. That is why I encapsulated nanosensors. I said, let's keep nanoparticles which can sense these changes, whatever those changes can be. Changes in pH, changes in oxygen concentration, changes in MMP biomarkers or whatever, right? We can have those things. The chemistry is quite known for that. So I have kept them so that I can get continuous readout by fluorescence, by electrochemistry, by many readouts. That's one thing. Second, very important part that we did was we miniaturized the system. So if you know, I'm sure you know about BACTEC, all these techniques which are used to detect bacteria in the clinic right now, they are all dyes. They use dyes which changes the CO2 level, and because of that there is a pH change, and basically those dyes changes due to the pH change, there is a fluorescence change, right? Though they give you vials. The smaller the vial, the larger the vial, more volumes you can do, more is the sensitivity. So by changing the vial sample size, you are increasing the sensitivity. Makes sense? More volume, more sample, more sensitive it is, right? We thought, now can we create a very small vial where the volumes will be, significant based on the size, right? So think of a micron size, so 100 micron bead. Right? In the 100 micron vial, even if I have, say, 2 bacteria, changing the pH because of the growth, changing to 4 bacteria in 20 minutes, that will be significant change in the pH. I'll be able to detect that. That's exactly what we did. We miniaturized the vial. We had a real-time sensor to see these changes, and there we go. It's a very simple solution that we have offered. We have not done any new things, just a small tweaking. And because we made it into micron size, it's microfluidic based. So there.
So that means I need to visit your lab on Monday.
Oh, absolutely. Please do. Please do. Yeah.
Yeah. Neetu, it was a fantastic talk. I missed the initial part, though I was in the other lecture theatre. So the thing is, the RBCs that you use, where are you sourcing them from? Yeah. And how do you, make sure that there are no batch-to-batch variations? Yeah. Right? And is it important to use autologous RBCs for the delivery?
Yeah, that's a very good question. And as I said, moving forward, we have so many things. So thank you for the compliment for the talk. The question that you asked about where are we sourcing it? So we are getting fresh blood from hospitals. We have a ethical approval for that. In fact, sometimes many of our students also go and get those samples. So we are using it. So we've done it with fresh blood, just so that we don't, worry about other things. But that is something we want to, we've tried with blood banks also, so we can get the nanoparticle made. That's nothing to do with the stability and the circulation time. We have not compared the circulation of these two different nano erythrocytes. That's something we need to do with fresh as well. So I would like to collaborate with some, maybe a proteomics person or someone like that who can, even if they tell me that the constituents are similar after, I think I don't have to do the in vivo studies then. So that would be an easier route, but the longer one would be to actually do an animal study on that. We have not done that. The question of autologous, yes, that's something which we are worried about. I've shown a very simplistic model of the blood, There are a lot of other things like the antigens will be present, and there are a lot of, absolutely. It has to be, right now I would say autologous. So it's a personalized delivery. It is, but it's going to be so difficult to translate because you won't have, It's not possible. So I am aware of that, but it's a proof of concept. But we are trying to see if we can, somehow take part of this and part of something like that, So we're thinking in that direction. We are aware. Let's see what happens.
The point, Taruna, you have raised about autologous RBCs, it's quite possible these days to come out with the individualistic kit, so wherein it can be done. But the follow-up kit has to be where it can be converted into.
Well, even if we do it for different blood groups, I think we'll be addressing some problem. But that also is going to be expensive. I'm not sure if the pharma companies would like that.
Don't worry about expense. If it brings value, expense is not much.
Okay, okay.
Any other questions? If no questions, let us thank Dr. Neetu Singh for the excellent talk. On behalf of organizing committee, the token of appreciation for Dr. Singh. So I invite co-chair Dr. Deepika to introduce the second speaker.
Thank you, Dr. Singh. Thank you, Dr. Neetu, for such an insightful talk and really awakening all our audience post-lunch session. I would like to introduce our next speaker in this session of medical devices, Dr. Abhijit Majumdar. Dr. Abhijit Majumdar received his PhD from Department of Chemical Engineering, IIT Kanpur, way back in 2010. His doctoral research work was on microfluidic assisted adhesion and soft mechanics. In 2010, he moved to INSTEM NCBS Bangalore, and then in 2012 to Harvard Medical School, Boston, for his postdoc research on cell mechanics and microfluidic devices. He joined Department of Chemical Engineering, IITB, in 2014, where he is presently working as an associate professor. He is an ex-DBT Wellcome Trust Fellow, TR35 India awardee, and also a selected member of Global Young Academy, Germany. Now I invite Dr. Abhijit Majumdar for his talk, please.
Yeah, great. But it is not coming. No, it is fine. It is going. Slide changer is not. Oh, okay. Thank you for this nice introduction, and thank you, Professor Sharma, Professor Dudal Panda, and all other organizing committee members. And as Neetu said, that we all would like to come to NIPER Mohali and be there. And this is my first time here, and yes, I mean, it's a beautiful campus. So I will be a little, the black sheep in the room, because as Neetu said that, she is a chemist and, I am a chemical engineer. So not from this pharma background, but, hopefully I will bring some flavor that you may find it interesting. Also, I must say that it is always a difficult job to give a talk after Neetu, because she sets the standard so high. But on the other hand, she also makes my life simpler, because she has given us some of these, the concept that I'm going to talk about. So as you can see that the title of my talk is this microfluidic approach. And then when I was searching, I was looking for some nice image and stuff in the Google, as we always do. Oh, I need to point that side, but it is not. So I got this one, this particular image. And then I actually kind of find, yeah, yes, this is a fantastic one. to talk about or to start with, because this is exactly kind of point out or kind of proof, put together what my lab is, working on. So the first one, as you can see the gradient, so the microfluidic devices, right? So as the name suggests, it's a fluidic device, so there will be some fluid flow involved, and it will be in the micron scale. And then these devices are used to create gradient of concentration. And why the gradient of concentration is required? Obviously, as you know, for the various different types of drug testing, or even to answer some of the basic biological questions, that we need to create, say, like the 100 micromolar, 50 micromolar, 25 micromolar, and so on. That we need to do manually in the lab. Many of the students sitting there, they know the pain that if they do a little mistake in their micropipetting, the whole experiment goes bad. Also, there is a loss due to evaporation which changes the concentration, etc. And so as a result, people create this microfluidic gradient device where you will have the flow of two or more say chemicals, and the output will be a concentration gradient which you can directly use for drug testing or any other testing and screening. So this is a bit. So as you can see, sorry, the aspect ratio got a little kind of disturbed. So this is one of such microfluidic devices that we have. I am not sure if the resolution is that great, but you can see that I am using two dyes here. One is red and another is yellow, and my output is a nice gradient of the color. So now you can put your output in your 96 OL plate or whatsoever, and I have shown here just 1, 2, 3, 4, 5, 6 outlet. You can have actually hundreds outlet in that system. But one thing is that in this one and most of the gradient making device that you see in the literature, and as the name suggests, they come with flow. And flow meaning you have to use some syringe pump, some connection, and most of the biologists, they hate that because that is another learning curve, right? So we thought of, say, can't we create something that will be a static device, just like your regular, the regular multi-well dish, or say like something like this in your Petri plate. You just add your drug at the source, and just because of the diffusion, it will create a nice gradient, and you can put your cells on all these spots so that they will experience different concentration of drugs. So this is one of the device I will talk about in details. This I will not talk about much because of the time. Second is the cell array. Again, as Neetu kind of showed in her presentation, that sometimes you want to arrange your cells, you want to pattern your cells, you want them to get aligned in a certain orientation. And this is something that we have again kind of developed in our lab. That the technique is called microcontact printing. It is not that we are the first one to do the microcontact printing. It is a very well established method. But then what we are doing, and for all of this and whatsoever I am going to talk about next, we keep two things in mind, and that is the kind of a motto of our lab. First of all, as again Neetu said, yesterday also some of the speakers said, to keep it simple. So we try to keep it simple. But other than that, we also want to keep in mind that we are in India, which is one of the low and middle income countries, LMICs. Can I create something that any and every lab can adapt? If I go by some hi-fi photolithography technique, maybe some of the universities and some of the others labs, they may not be able to do. So what we try to do in all of our technology part, there we try to do things something like do it yourself, DIY kind of technique, that just whatsoever is available in the lab, with that you can create something. So this is these are the protein dots, what you can see here, and where you can put your cells, or you can do some the antibody antigen kind of testing, and here you can see the cellular alignment. Again, this has not been done using any photolithography technique, something like the beads, capillary tubes, and the syringe that is available in the lab. So if any of you are interested, I can talk about that, as a one-to-one discussion. Third one is the organ on chip. Again, as I said, that after Pitu it is difficult, but also it is easy because she has already introduced you all of these concepts, right? So if you want to go for a different type of drug testing, nowadays, various countries, they are kind of putting ban or making the animal testing very kind of restricted. So it is getting more and more difficult. And that's why the field is also trying to go into, towards that if we can mimic the organ and can at least our in vitro testing can be. And actually people talk about that it is not alternative to animal testing, but people talk it about alternative to human testing. And then they say that what we check in the animal, say the small animal in the mouse, right? Most of the result, like 10 drug that gives you a positive result in rat or mouse, that the 9 out of the 10 fails in the clinical trial. And there is a saying is that human is not a 60 kg mouse, right? So that's why we would like to create is something like called the organ on chip, where we use human cells and we try to recreate some of the organs and then kind of which can be used. So it is not artificial organ. This is for drug testing, drug screening purposes. So here I have one design that is the skin on chip, which creates a air liquid interface. That is done in collaboration with Dr. Prajakta Dandekar from ICT Mumbai. Here we have a retina on chip where we are culturing retina cells as well as the precursors. Again, this is in collaboration with Prajakta. This is not our design. We have copied it from a paper, but we are now working on this and we are further modifying this. This is a placenta on chip and working with Dr. Deepak Modi, Teruma's colleague. So with him, I am working on this. So these are the few things, and then another thing that we are making, and that is spheroid making device. Again, Pitu talked about the importance of working with spheroid and the tumoroid, and this is again a device that we are making. You can see the size. She also talked about the importance of miniaturization, so we are miniaturizing it. And from a small plate, you can get many spheroids or tumoroids or organoids. So this is something that our lab is doing, and that's why we call our lab as AIM lab. AIM does not for Majumdar lab, but AIM stands for mechanobiology. That is what we are looking at, the effect of mechanics on cellular behavior, on stem cell and cancer. AIM stands for matrix and materials, so we are looking into the effect of various materials on cellular behavior. And the last AIM obviously stands for microfluidics. So that is what I will be kind of talking more here. So today I will kind of talk about this one technology where we make spheroid, and this one gradient one. So first I will start with the spheroid one. So as we were discussing that this is the kind of thing that we all do, that the 2D model in vitro, and then going for the animal model. Both of this model have their huge advantages, obviously, that's why it is there for so many years, but they have their disadvantages as well. And as I have mentioned, lot of the things that the result that we get in vitro or in the animal model, that cannot be translated when you test in the human. And that is one of the kind of very big limitation, that our present model systems are not predictive enough to kind of tell us what is happening inside our body. And one is that here we are working in 2D, where all the cell contact, 3D structure, gradient in the penetration of the oxygen, all are missing. So that's why we would like to create the 3D model, and probably some of you have already worked on the spheroids or the tumoroids models. So if you have worked with the spheroid and the tumoroid models, you know that the various method that is used to make the spheroids, one of the most common is the hanging drop. that you put the drop on, on the lid of a petri dish, then you take it upside down, and because of the gravity, cells get, accumulated. It has two major problems. Number one is that it is a laborious process. It cannot be high throughput. And second is that because of the evaporation, there is this tension, and it creates the osmotic tension, which causes some cell death. And you really cannot, make it bigger. So you really cannot grow it further. There are other methods, such as people use ultra low adhesion plates. But again, those plates are quite costly, first of all. And if you think of that, if you want to create, say, you take a 384 well plates, you will create 384 spheroids by adding cell and media individually in those 384 wells. So what we thought is that, can't we make the life a bit simpler? And as I said, we always work for this do-it-yourself kind of thing. So here what we have made is that we took a 384 well plate and we miniaturized it. How, I will talk about in a bit, but you can clearly see this is the kind of size. So two steps of miniaturization, we call it S1 and S2. S1 size, each of these goes into one well of a 6 well plate. It is 10 by 10. So from each well, you get 100 spheroids, and from one 6 well plate, you get 600 spheroids. And if you go to S2, the further miniaturization, you can get 1200 spheroids from one device. And this is the reduction in the quantity of the material, or the reagent, etc., that you are using per well. And as again she was suggesting, that there is a benefit of reducing the total volume. So how we are doing it? So I first give the credit to these three people. Pankaj, he did his PhD with me. Now he is doing postdoc at Duke University. Saurav just joined as a postdoc, and Tushar completed his master's with me. So these three people actually worked on this whole thing. I will not go into the details, but I will give you a very quick idea that what we are doing. So as I said, we work on something to make life simpler, and something what any lab can do. So we took a 384 well plate, took it upside down. So if you look from the from the bottom, you actually get some hills. Then you put gel, polyacrylamide gel available in any microbiology or the molecular biology lab. We put that polyacrylamide gel so that these hills will now become small wells. But then in this PA gel, you can shrink them, because if you dry them, they will shrink in size. So we dry them, although it sounds very kind of easy, but drying process is not that easy, because you have to keep it absolutely flat and no distortion. But anyway, we have worked on that. So you can get a very, a smaller version of your 384 well plate. And then you do it multiple times to get into the other smaller structure, and finally you can come to this level. So I'm just kind of carrying just a couple of these ones to convince you that yes, we have really made it. And if any of you are interested, you can just have a look at these ones. So okay, so after we make it, so what do we do with this, right? So yeah, so this is exactly what I was talking about. I mean, again, all the aspect ratios got changed, but this is, if this is your size of a 384 well plate, after first shrinkage, this is the size, and after the second shrinkage, this is the size, and this is the kind of well you got. So you put your cells and you get your spheroids or the tumoroids, whatever you call it, organoid, etc. So as I said, that if you have a 384 well plate, as a PhD student, if you want to make the spheroid, you have to add media and cell in each of them individually, right? Here, because of the miniaturization, you just can add your media like this at one shot, and you will get 100 spheroids ready, okay? And So this is essentially you put the cells and they get just collected, give you the nice spheroids. And during harvest, to harvest, that is also easy. You just need to turn it, flush it, and you will, you can collect all your 100 spheroids at one go. So this is something just to show that yes, those spheroids are live. So this is a live dead assay with calcium. And this is just to show the some of the, the spheroid growth rate, that from the day 3 you get your first spheroid that you can look at and how they are growing. And we are comparing that with the ultra low adhesion plate that is, most of the labs use. Also, one question comes in terms of what is your distribution, size distribution, because you want monodispersed spheroids, right? So as you can see here, the red one is your ultra low adhesion plate spheroid size distribution, which is the gold standard. Our S1, that is the larger device, that gives a little, the distribution is wider. But if you go for our S2, that is the 12 well plate device, you can see actually tighter size distribution. So you get monodispersed 1200 spheroids at one go, and that you can make, in your lab yourself. You don't even need to come to me or buy it. And this is just to check at the well occupancy, because the question comes that you are just, putting at one go. Are you getting spheroids in all the wells? So yes, out of the 100 wells, we get 88 to 92, that kind of number we get. So the 90% wells, we get the spheroids. So then the next, we went to check for the drug testing. So we used multiple different cells and the drug combination. Here I am showing you for the glioblastoma cell line U87NG with temozolomide and jar cells with EGCG. And then we are comparing again the gold standard ULA plate and the S2 device, and we are trying to find the IC50 value. You need to understand that you can easily collect your media from here, so you can do your MTT assay. Also, because the material is PDMS, which is transparent, you can also do the imaging without much difficulty. So this is the various different drug and the cell combination that we have tried, and you can see that these numbers are matching well with the gold standard, that is ultra low adhesion plates. Next we thought is that, okay, fine, the spheroids are made, but the student needs to come to the lab every alternate day to change the media, right? So can we reduce their work further? And this is again in the same device, we just added, it is absolutely simple, we just added an inlet and outlet, and with a pump, with a syringe pump, and then with, so you can see that this is in the static device, that is where you need to come to the lab to change the media, and this is the flow device where you don't need to come to the lab to change the media. In fact, there you get more number of spheroids because you don't lose them during the media change process. So essentially you come to the lab, you take this device, you put your cells, go home, come after 7 days, all your spheroids are ready, you harvest them and start your work. So this is the first one that I was talking about. So we have a static device, we have a flow device, and it is economic, easy to use, easy to handle, and where I feel that the most important part here is that anyone can make it in their lab. So it is not patented, nothing, you can just do it yourself. So this was the first one that I wanted to talk about. So how much time do I have? 10 minutes, right? Yeah. So the second one that I wanted to talk about was about our that gradient making device. As I was talking about, that for drug testing, you need to create gradient. Again, you do that in your 384 well plate by painstakingly adding your drug or your compound at different amount. So that we wanted to, again, to make a concentration gradient there. This is what people do, but this is time consuming and erroneous. As I said, there are the loss due to evaporation, which changes the concentration. Options, obviously the robotic arms are the options that is taken in the pharma industry, but that comes in crores, and not many of our labs can kind of, spend that kind of money. Another thing that I said that is quite often, and you will see many papers, that is this microfluidic devices, where they have a flow and that creates the gradient. But as I said, they need, they need lot of these accessories, and I have, spoken to many biologists, and I know that they don't want to get into that kind of, because that is, again, that is not that easy to kind of handle all these things. So we thought that, okay, can we have a device? And also, you need to remember, when you add these tubings, valves, pumps, etc., it is no more portable. You cannot take it from one place to the other, right? And if there is any loose connection, etc., there will be leakage and, lot of these issues. So we thought, can we get rid of this flow business? And so for that we created something of this design. So this is done in collaboration with Professor Prasanna Gandhi in the Mechanical Engineering Department, IIT Bombay, and work done by these two students, Ketki and Shital. So this is the device. So what is this device all about? So can you please run this video? Excuse me? Can you please run this video? Yeah. Click. No, click. It should work. I mean, just click on that one. It is not working. Okay. Now you have to trust me. I cannot show you the video. So, so can you please exit and try it out? Exit the PowerPoint mode, presentation mode. Now can you please run it? No, I don't know if it is in that. Okay, fine. Anyway, so please go to a full screen mode. So again, as I was telling you, that we wanted to make things that anyone and everyone can do. So we wanted to, first I will tell you what is this device about. Yeah, can you please go to the presentation mode? Yeah, thank you. So I will tell you what is all this device is about. So this is essentially some microchannels connected, nothing more than that, nothing big deal. Where I have a sink and I have a source, so two reservoir at two ends. Okay. Where is the beauty? And this, where these microchannels they intersect with each other, that we call as node. Okay. So no big deal, right? So what is the beauty here? Beauty here is that if you look at the cross section where the nodes are of this kind of dome shaped. So now when you, what we are doing here, so we have these connected channels, we have a source, we have a sink. First you fill everything with media, then you add cells, the way you can see here, you add the cells. When you are adding the cell and the media, due to this dome shaped, so there is a change in the, so some fluid mechanics is involved here, that it is like the flow was going, all of a sudden the cross sectional area increases. So you can think of when a river flows some sand, if the cross section increases, its velocity suddenly goes down and it deposits that sand. Exactly the same thing, when the flow comes to this node, their velocity goes down, they deposit the cells. So in each of these, say if I have this 5 by 5, so 25 nodes, or 10 by 10, 100 nodes, in each of these nodes you will get cells deposited and your cells will get adhered there. So it's a view, top view of the node, and you get the cells adhered there. This kind of shows that it is a 5 by 5 25 node device. It is 10 by 10 100 node device. So you can see that in each of your node you have kind of equal distribution of the cells. Once it is done, then you add in your source, you add your drug, and by the simple diffusion process there will be, so here I am, I have added FITC. So as a simple diffusion process, in each of this node you will have the different concentration of the drugs. So the cells sitting here, they will experience higher concentration compared to cells sitting here. And as I said, that because it is based on the glass, you can easily image them. So the first question that we asked, is my gradient stable? Because if it is changing, then I really cannot do much. So we get a nice stable gradient that you can see here. Again, some mass transfer concept is there that we had to optimize in terms of length and the cross-sectional area of the channel. And then we did some computational simulation to show that the gradient of the for the different time point. Next, what we do, so it is just a proof of concept. So you take two devices, one is your control, another is your test. You put the cells in both the devices. Then in your control, obviously you don't add any drug, but in your test device, in the source end, you add your drug. And then for each of this node, you get the different concentration of drug. Close to the source, drug concentration is the highest. Furthest from the source, drug concentration is the lowest. So you have more number of cells here. Cell survival is more. There is, so the more cell death. So after 24 or 44 hours, depending on what drug, what cells you are working on, you just take the image, put it in the computer, put it into any image analysis, image kind of software, and that will give you the cell number and cell number per node. You already know the concentration in your, so I say for example, I see here at node 3 I have 50% cell. You know the concentration, and that is your IC50 value. So that we have again checked with a standard method of the multi-well plate assay, and you can see that the number kind of matches. So we have done for other combination, so I am not going there in the interest of time. Another good thing is that once your experiment is done, you can actually get rid of your so again the size is something like this. I don't know if it is visible for you, so something like this, and once your experiment is done, you can get rid of that PDMS mold, but your cells are already adhered in their position, so you can fix them and then you can stain them for the post drug treatment assay. If I have a collaborator like Nitu, I can also put the sensor in each of the node and I can get the real time data. So so I have just shown you the one directional work, but you can now think of in your imagination, this work is going on. You can actually now test the drug combination. That is what we are doing with CMC Velour with some patient sample to look at the drug combination. You can go to 2 drugs, 3 drugs, 4 drugs, and you can change the design to even make it further, the factorial designs you can do here, exactly. So, so my take-home message, I am kind of at the end. So my take-home message here is that this is what we all are doing, and for our lab, our mantra is to keep it simple. And so that, as I said, we have 2 vertical where we are asking the fundamental questions, with the mechanobiology. There we are asking, there we don't care about it is simple or complex. But when we are developing the technology, our first goal is we have to make it simple, we have to make it affordable, and we have to come up with a design that anyone can make in their lab. And this is what I have shown you. I hope that Some of you may be interested and you can just kind of adapt that in your lab. So finally, to give the, the credit where it is due, both of the work was funded by Imprint 1 and 2. My lab started with the DBT Wellcome Trust of funding as well as funding from IIT Bombay. I cannot be thankful enough for that. Vadvani Research Center and the DST SARP funded other projects that I have not shown here. Today's work, one of the work was done in collaboration with Professor Prasanna Gandhi. These are all the students who did all the hard work. Again, as she said, we just present and take the credit. They do everything. And this was the group before pandemic, and hopefully it will change. And yeah, this is the group after pandemic. So thank you, thank you for your attention. And I'll be happy to take further questions.
Thank you, Dr. Majumdar. So the take-home message we can have is, the mantra is to keep it simple.
To keep it simple.
Okay, wonderful.
Kiss.
Now the house is open for questions.
Great work, Abhijit.
Thank you.
My question is on your first part where you're trying to make it simple for this Freud preparation. And, you are developing it by shrinking the material to a certain extent. And then it maintains the well, where the cells randomly goes. So there the control in cell number is not possible, in my opinion.
So that's what, we have shown, that it is in a size, and if you just kind of put it like this, you are not very precisely controlling the cell number, but final spheroid, spheroid distribution that you get, that size is a narrow distribution.
Does it affect on the time, at what time the spheroid is fully prepared?
So that depends on two factors. That's a very good question. That depends on two factors. Obviously, your initial seeding density, like what number of cells you are seeding, and whether you are using S1 or S2 device. Because if you make the S2 device, because it is smaller, it kind of brings the cells closer quickly, so you get your spheroid quicker in your smaller one.
So there will be time differences.
There will be the time, but you will not work with S1 and S2 together, right? You can,
I know.
You can decide which one you want.
And the second question is on the retrieval part, that you said you simply flush it and tilt it and probably they will come out. Is there any capillary effect we have observed? Capillary trap, like they are not coming off? And
no. Okay. Because I mean I have not talked about that. So this device is made of PDMS. We coat that with pluronic to avoid cell adhesion.
All right.
So you just do it and they come off easily.
Good. Okay. Thanks.
Thank you. Yeah. So for the spheroid, as I have shown, we have used multiple different cell lines. We have used HMSCs, glioblastoma, SHSY5OI, C2C12, JAR, BIVO, HeLa. So we have used like 8-10 different cell lines to make spheroids and various different drugs also.
Yeah. And my next question is, what is the particularly dimension of your well, sir? Particularly the depth of the well.
So I was, it was there, I don't recall exactly. Volume wise, the smaller one is 1 microliter each well, and the S1 is 5 microliter, S2, I need to check that. 5 microliter and the 2 microliter, I guess, or 1 microliter.
And last question is, how you looked at the spheroids from the well, sir?
Retrieved.
Yes.
Yeah, so that is what I was exactly saying, that retrieving is absolutely easy. You just tilt it, flush it, and they come together.
Thank you, sir.
Because many of the downstream experiments were done after retrieving those spheroids. Yeah.
Outstanding talk. Okay, you come here for one good talk, you get 2 good talks. So buy one, get one free. Kind of.
Thank you.
So my question is for the second part. Okay. I am a microbiologist, so I am interested in testing new drug or new antibiotics. So the question is, how can you control the concentration of each node for the gradient, or can you do that, like to test the MIC values or checkerboard concentration and so on and so forth?
Yeah. So for the microbiology kind of application, I mean that's a shortcoming at this moment, because this work with the adherent cells. If my cells are not adherent, then it will not work, right? So that's a kind of a, but we can modify that, something we are thinking of. But coming to your question, that the concentration, so you add your drug concentration, you determine that, you put that in the source. And because of the diffusion, so that we also have done the simulation, as well as we have done with FITC and the FITC dextran, of various molecular weight, that within 6 hours, it takes around 6 hours to get the gradient. Once the gradient is set, what we have seen that it is, it is a constant for like 5 days. It is, quite stable. If you want, so and the different nodes has different concentration. If you now want to change that value altogether, you need to add more drug in your source, and that will shift the curve.
Adherence cell probably is not a problem, because you can make the biofilm first and then put your whatever the node and do that. Have you tried that?
So we have not tried that actually, but because my lab does not work with that, but I was talking to someone and actually we got that suggestion that yes, this can be done. So that is something, yes. And actually that takes me to one more thing. Before I forget that, I mean, again, this is for the students, that the Ketki, when, she was working with this device, so if you remember, the cross section of the node is dome shaped, right? One day, just by mistake, she put her device upside down. Okay, so now the domes, earlier cells were supposed to sit on the glass bottom. Now cells were sitting in those domes, and what she got is that spheroid in each of the dome. And now she can do the drug testing on spheroids using the same device. So that is just because she was playing around, she got something interesting. I think the time is.
Sir, just I would like to have one short question regarding that dome. Like, how are you fabricating it? How you are maintaining the unique height of the dome?
That credit absolutely goes to Professor Prasanna Gandhi's lab. I could not show you the technique. Do I have some time to describe the technique? Because that is a very interesting technique that they have developed. We are just using it. So you take 2 plates, okay, 2 acrylic sheet. Then you put some ceramic material. You can think of, just imagine your toothpaste, Colgate toothpaste there. And then when you separate them, it is called something called Safman-Taylor instability, because the air now going in, that displaces the liquid in a particular structure. Okay? And that structure is very well defined by the physics of the system. If you control the viscosity of your fluid, surface tension of your fluid, and your separation velocity, your things will be absolutely standardized. What they have done a very fantastic thing, is that, so that if you just separate these 2 plates, air is coming from, from the circumference. What they have done, in one plate, they have created holes at the specific location. Now air is coming through those holes, and now by designing where to put the holes, you can design that what will be your final structure. So as I said, again, it is a do-it-yourself scheme. You just can make the holes in your workshop. It can be done. The paper is out that what should be the dimension and etc. And anyone can make it. So the dome structure, etc., the width, length of the channel, etc., is very, very, very well controlled by the physics of the system, because the physics is so well established.
Thank you.
Thank you. Thank you. Yeah.
Exactly. So I don't see, but the mind doesn't know, Thank you so much and making us enlightened about the systems. Yes, sir. Sir? So now we have come to the last session, the last talk of this session. This is by Dr. Sanjeev Kumar Gupta. Let me his introduction was really very interesting. Let me speak about him. Dr. Sanjeev Gupta is a QMS, Quality Management System person, regulatory technical professional with over 2 decades of experience in the field of medical devices and in vitro diagnostics. He holds a doctorate in biotechnology from IIT Delhi and CSIR IGIB and a postdoc fellow from Oxford University, UK. He has held leadership roles in several national and international pharmaceutical companies with focus on all the classes, Class A to Class D, of medical devices and in vitro diagnostic products. He has successfully transformed the quality management system processes in these companies. He is also credited for bringing first ever WHO pre-qualification of malaria rapid test. He started in Trust Consulting in 2020 and is currently working with several companies in India and abroad. He's also a consultant to WHO as well as involved in WHO JSS PHARMACON mentorship program. He's also running
This is
an
forward.
NGO for psoriasis and autoimmune skin disease. He has written 2 books, several relevant CME articles, and health articles for several newspapers. So I welcome Dr. Sanjeev.
Thank you.
for his talk.
Thank you. Thank you, ma'am. Did you get the PowerPoint? So good afternoon, everyone. By the time they get the slides on board, I'm so happy to be here along with IITians Delhi and Mumbai. And I was in IIT Roorkee some 25 years ago. I never thought I would be doing these kinds of things when I finished my master's and my doctorate. But that's the way it just works. That's not the first slide, I guess. Sir, your screen is spoiling the slide, sir. No, even that's not the first slide. No? Is it? First slide is the title. What have you done? Anyway, we can start from here. Can you please put this into? No. There are 3, but you can pick up this only. PDF. No. NIPER talk. You are at the right. Can you check the room? यही है क्या? आई एम डाउटफुल. ओके तरुणा जी प्लीज प्रोजेक्ट इट फ्रॉम हियर ओनली. ये डाल दीजिए आप, कोई बात नहीं. स्लाइड प्रेजेंटेशन मोड. Okay, sorry for the disruption in the slide presentation, but this slide tells you, basically tells you my journey.
which started in 1995. I started with a pharmaceutical company, a very small company in Faridabad, and in 2020 I started my own consulting firm. In between, I worked with the three multinational companies, which is Alair, Abbott, and Arcray. And I've been working with the diagnostics and medical devices, and currently I've been working for about 18 companies, and Tata Steel being one of the largest client which I have, working with different materials. And I've been also working with a couple of startups, and I've also done some pro bono for different startups. So I look at things a little differently. Our company is known as In Trust, basically. It's something which has just come on its own, that you are in trust, basically. And this logo reflects three important things in any process. The one actually is a square. This is called starting a process. The one in the green color is completion of the process. And there is a diamond part, which is called decision making. And these spokes are reflecting continual improvements. So it's basically a process. Nothing works without this process in industry. In academia, it works a little differently. A couple of my consultancy areas are about infrastructures. We design laboratories, we design companies, manufacturing units, quality management system, and quality culture in organization, especially about design and implementation. And we do a lot of training for this area. And the technical includes the clinical investigations, essential principles, risk management, stability studies, labeling, performance evaluation, and clinical performance evaluation. I'm sure many of you would not even know these words, because that is the language of regulation. If you really want to understand what a regulation is all about, you need to get the regulatory language and terminology right into your projects, into your working, into your conversations. A lot of training which we have done during COVID times online, and we've now started doing personalized trainings. This particular slide includes, I'm sorry, this quality has gone really bad. This particular slide is about when I trained last year, about 80 companies on behalf of Association of Diagnostic Manufacturers of India. So this is, today what I'm going to be touching upon is that development of in vitro diagnostics medical devices, role of standards and monographs. Why I've chosen this standard and monographs is because it connects with the audience and pharmaceutical segment. They know monographs. That's something which they know. Any pharmacopoeia, monograph. Anything, standard. Okay. So this is very important, that what is to be considered when we develop an in vitro diagnostic test. There are different kinds of in vitro diagnostic tests. If you really look at it technology wise, there are lateral flow devices, we all know, COVID antigen rapid test. Then we have a flow through device, wherein like rapid flows like immunochromatography, whereas flow through is gravimetric flow of this thing. Then we have a lot of molecular tests coming, urine chemistry, and a lot of machines are there, you can see, which are machine based tests. And you have, this is a beautiful product which I launched in India when I was working for Alair. This is a miniaturized PCR machine which can work in snow laden hills to Rajasthan, where 45-degree temperature is there. This was for HIV test. It's a very beautiful machine. Unfortunately, it is discontinued after the company was sold to Abbott. So giving you a brief about this is that once we talk about the regulation, particularly about Indian regulation, there is a rule 7 in the regulation. Our entire rules are divided into chapters, schedules, and rules. Okay. So rule 7 talks about the product standards for medical devices. So what does it say? What the regulation is telling any person who wants to develop a medical device, that follow Indian standards. When they say Indian standards, it means Bureau of Indian Standards. The mark which you word ISI. Okay. So if they have a guideline, follow it. If they don't have that guideline, follow any of the ISO standards. ISO standards are generally for the European countries, wherein you have 29 EU countries. If they also don't have something, go to US, follow ASTM standards. If nothing is present, follow your own standard. Okay? So your own standard comes at last. Unfortunately, in academia, this comes first. It's reverse. And that is why many products don't see the light of the day. Okay? So what needs to be understood is this. I've been for last 2 years, I've been very vocal on every platform how the institution should be working. Okay? Then there are horizontal standards like ISO 13485 2016. It only tells you how to organize. It's a horizontal standard. There is another standard called 14971. It's a risk management standard. It's again a horizontal standard. It's generic in nature. Horizontal means generic in nature. Then you have this biocompatibility standard 10993. A lot of people talk about 10993. They hardly open it. They hardly open it. Because why we talk about biocompatibility, but what biocompatibility test has to be selected? Unless you go through step by step, you will not be selecting the right test. If you have not selected the right test and you go and submit your document, the guy sitting on the other side of the table knows what is to be read and how is to be read. He'll say, whatever is necessary, you haven't done it. If you haven't done it, your all 3 years, 4 years, 5 years is gone waste. Okay? Now, between the standards and monographs, I want to tell you, there are certain overlaps. Like blood grouping reagents, they are covered in Indian Pharmacopoeia 3290. But they are in vitro diagnostics, they are also covered into medical device rules or CE marking or 510K, everything. If you look at a couple of anti-HCV, anti-HIV, HBsAg and other tests, there are CDSC has decided the guidelines. These are neither published by Bureau of Indian Standards, nor they are published into any pharmacopoeia, but then CDSC has decided it. Where does the CDSC get it? So we are very good. So we copy it from EU. So we copied it from EU. Or we copy it from US. There is something new which is coming up. It's called WHO Technical Standard Series. This is the most toughest one, most complex one, and something which really makes your products pretty big. Now, a lot of people talk about stability studies, right? In pharmaceuticals, they generally say ICH guidelines, right? Well, it doesn't apply into medical device and IVD, but I've seen many people talking about during their R&D also in companies also that we apply ICH guideline. It doesn't apply on you. Why do you apply it? So all your studies, all your data has gone waste, right? So if you're working with an in vitro diagnostic medical device, there is an ISO standard called 23640. You design all your experiments using this standard and you will get everything right and regulation will accept it. But if you now bring in ICH here, you're not doing justice. Same way we have EP25A for stability studies, you choose any. Same way we have WHO guidelines, right? Which is again, TGS2 is having connection to these two. So selection of a right standard is very, very important. Now what do we detect in a diagnostic test is a very big question for many people. And many a times there are not clear-cut answers. I've tried to put something with examples here. We may be detecting an antibody, which may be IgG, IgE, IgM, IgA, any type, okay? We may be doing a protein antigen, which is like HBsAg, prostate-specific antigen, or any of the fungal antigens. Any other protein marker which does not fit in here. Antigen isoforms, particularly in prostate-specific antigen. A glycan antigen, we are looking into cholera. This morning we had a lecture. A glycoprotein antigen, again, in cancer, all cancer markers are generally glycoproteins because they have repeated structures, tandem repeats, along with a good amount of glycosylation. A hormone or its derivative or its metabolite, any of these. Gene targets when it comes to molecular diagnostics, gene mutations when it comes to molecular diagnostics, and any other biomolecule. So there's a list is large. What we really need to know is what exactly we are detecting. If that is not defined well, your product is rejected by the regulation. So what I'm going to tell you is that in view of this, as to what we are detecting, I picked up one example. This is about TB and resistance detection. Unfortunately, slides have really gone bad. I'm not happy speaking what I'm speaking. So there are 4 or 5 different kinds of products in the market. Who detects what? So these are the number of markers which are being detected. These are the gene targets. And this one is for rifampicin. This is for isoniazid. I think this is also for isoniazid. So there are different technology platforms in IVD. Why I'm showing this slide is because requirements for telling that my product is good varies from technology to technology. More complex the technology, the more data is needed. Okay? If you want to look at from the product type, it could be simply a reagent, instrument, accessories, or everything put together. It could be a software. A lot of people are confused about softwares. Everyone says I have a software. It's an IVD product. It's not. Every software is not an IVD. Tech platform, it could be an immunoassay, hematology assay, clinical chemistry, microbiology, or a molecular assay. Disease type, if you look at infectious, cancer, cardiac, autoimmune, nephrology, GI, and others, we must know categorically. Then user type, it could be a self-use, used by a technician. In a path lab, you use it by machine, or a hospital, or a point of care test. There is a very big difference between self-test and point of care test, and many a times, some people will say, my product is for lab test also and self-test also. Well, you cannot make this claim unless you have clinically proven that it has been tested in a clinical as a self-test product. It has also been tested in the clinic as a clinical product. So data is different. Few examples of IVD products where I've been involved in last 25 years. I picked up a couple of them. This is one of the paper which was done in International Journal of Biological Macromolecules. I'm sitting here. This is what we did, development of glycan-specific lectin-based immunoassay for detection of prostate-specific antigen. One of the beautiful paper we had, wherein we were working on different isoforms of PSA, and we tried to set up an assay. And I left the company, so I don't really know where the assay stands now. This is Taruna's technology. She's sitting here. And it just happened so that when she wanted to do validation of it, I was working in Span Diagnostics, that I made the validated kits in Surat, and we did some of the stabilization works and other things. This is now probably up for sale at ICMR website. This is something which we did before, a year before the COVID, where we were talking about multi-influenza rapid diagnostic test. The important thing is, I've told many friends that influenza committed suicide after COVID came in. You hardly see influenza cases, but that's what is reality. Okay, so this was a rapid test which could detect influenza A, B, and H3N2 and H1 differential diagnosis through a rapid test. This is the fastest development we finished in 7 months. It was tested in skims, but by the time we came to the launch of it, the COVID had struck. So this product has now taken a backseat because now combination products are coming. This was a team which worked on this. This is the most recent one which we have done for one of my client company. I've been involved right from the beginning, and bringing regulation, QMS, and everything into this product. You can see me holding these kits here. This is a molecular test for detection of TB and resistance to isoniazid and rifampicin in one go. Existing tests which are there in the world, I'll come to a little later to that. Existing tests are separate tests, and this one is the one which goes together. So this is a beautiful outcome, and it has taken a lot of time, but I'll show you some good outcome which is there. On a couple of slides, how we should be working, whether it's a research organization or a company. Basically, we first need to know what is the user need. If we have not defined the user need, you can never define the regulation. Okay? Many a times what happens is that institutions, whether it's IIT or ICMR or any university or NIPER, you generate clinical evidence. That's it. But you think you have developed a product. That's not correct. It's just a clinical evidence. There is something which has a potential to become a product. Okay? And what brings a technology to make a product is so many things. It's not whatever good amount of science you have done. If as an industrialist I don't see a potential of revenue generation, it's just a good science, nothing else. Okay? So, but nevertheless, we should understand what is a user need. That's the most important, I think. So, if I look at TB, a single molecular test, please remember this word single, a single molecular test which can detect TB and resistance to frontline TB drugs, isoniazid and rifampicin, can be very helpful in TB programs. What is the TB program? National TB Elimination Program in India. It is globally. Okay? WHO wants to eliminate TB by 2030. Indian government wants to eliminate it by 2025. It should be affordable. Should be easy to use. Very simple term, easy to use. But if you bifurcate this into 10 or 20, you will understand where you stand. Point of care, huge confusion about this. What exactly is point of care? So from the user needs, we convert it into a design input. Please remember the terminologies. User need, you hardly use in your research activity, user meet. Clinical evidence, you hardly use this word into your documents. Then design inputs, you will never use this word called design inputs because it's a regulatory requirement. And you imagine when you present your data as a design input, the reviewer on the other side of the table connects. Okay, that's very important. So when it comes to in vitro diagnostic products, couple of inputs which we have to take, okay, is very important. Sensitivity, one of the most confusing word for many people. Why? I will try to tell you. There are two kinds of sensitivity. One is called analytical sensitivity, other one is called clinical sensitivity. Most of the papers which are published from academia, they don't say whether it's clinical sensitivity or analytical sensitivity. They just say sensitivity. Okay? When they just say sensitivity, it is incomplete. You don't know what's happening. So in the analytical sensitivity, which is also called detection limit, okay, there could be a quantitative analytical sensitivity, there could be qualitative analytical sensitivity. Then there is when we talk about clinical sensitivity, it always has to be against a gold standard reference method, where we are talking about accuracy and trueness. Okay, that's very, very important aspect, and it has to be presented properly into a 2x2 table when you have worked it out. Most important thing again is that we really define right in the beginning what we are looking at as analytical sensitivity, what we are looking into clinical sensitivity. Same way we have analytical specificity, then we have clinical specificity. These are important terminologies. If it is not presented properly, your test is not going ahead. Then we look at reproducibility. You know, different people will look at reproducibility in a different way. So we have between different operators within the same day, between different days, between different batches, between different instruments. Why is it? Bring variability and see whether there is an impact. Recently I was reviewing a submission by a manufacturer who submitted the product for WHO pre-qualification, and we found that they have miserably failed in this. So there are 12 months of work has gone waste. Repeatability is about uniformity. Dr. Majumdar was telling about different wells. So when repeatability means if I take one sample and if I put it in 96 wells of an ELISA plate, that I get the same value if everything is under control. Okay? So that's easier said than done. Pipetting variations, coating variations, dilution variations, okay, reading variations, how you do it. That's why things go mechanical, because then you have very quick time through which it is happening. When you do it manually, you actually end up doing a lot of things wrong. Okay? And that's where the development comes after you have designed something. Linearity. You want to see a straight line, you want to see a little semi-curve, you want to see something like this. And when linearity also decides whether your in vitro diagnostic test is going to have a hook effect. Many people don't look at hook effect. It's a very important segment of the characteristics of in vitro diagnostics. So these are the three molecular tests connecting back to TB molecular test. This is worldwide sold. This is made by Danaher, Cepheid. This is a cartridge-based test. This is a laboratory-based test. You need a high-end laboratory for this. This was superbly challenged by an Indian company called Molbio. Today they are a 1200 crores company. They are based in Goa. What they have done is they have miniaturized this, and these two can be taken to any part of the country. You can take it to a village and do the TB screening. Again, cost is one big deal. Okay? But then you also have something called line probe assays. Line probe assays are lowest form of it, but that plays a very significant role into TB diagnostics. So when we started looking at this TB product, which I'm going to talk about into this, is that we were having a PCR reagent kit and a rapid test device. One will do amplification of the gene targets, and the other one will see how it is read. Okay? We took inputs. Please remember design inputs. Okay? So we started looking at, for TB diagnostic, the global reference is only taken from WHO. WHO gives this reference to the whole world. So you cannot say that I took it from Hinduja Hospital, I took it from there, I took it from there. Globally, it has to be WHO. And WHO has a consolidated guideline on tuberculosis diagnostics. Okay? So with that, once we start talking about design input, this is the first design input that we take. Then we also decide, well in the ahead of product completion, what all kinds of stability studies we'll do. Like in pharmaceutical, you do stability studies of your product, but in diagnostics, you also do the stability studies of the specimen which you are going to use. Blood, serum, urine, sputum. What is the stability of that specimen so that you are diagnosing the right thing. So that means among various kinds of stability, you also have to do the specimen stability. And these are all difficult ones. These are not easy ones. Same formula doesn't work for everything. So you have got a user need, you've got a standard, you've got a design input. Now you're going to start your R&D activities. And then once you do your R&D activities, so we normally call it design and development, you all call it R&D. This is the regulatory terminology. We have to draw every flow chart, so what you want to achieve. If you don't have it, it would be like your PhD program that you start with the synopsis and the objectives are changed at the end of 5 years. Okay, when you are developing a product, if you land there, that means you were either wrong in the beginning or in the process or at the end of it. You can't change when it comes to the product. You can when it comes to MTech or MPharm or PhD thesis. Okay, so this is very critical for us in industry when we work this. Everything needs to be worked out. And you see, I was telling you in my logo, this is a decision making. Okay, diamond. So we standardize everything for this particular molecular aspect of this kit. There were 2 kits. So the multiple rounds of experiments were done and everything was fixed. And then we also decided how we are going to be testing at each level. And then the biggest challenge was to fix up the cassette. There's no one in India who makes these kinds of cassettes. So we developed, and you will see that there are a lot of slides which we really worked with in terms of setting up. Why we are doing so much of work? So many regulatory people and some even doctors think that it's a packing material. It's not a packing material. It's part of the formulation. If you get this wrong, you lose your analytical sensitivity, specificity, clinical, everything can go for a 6. So that is why it is a part of the formulation, like a buffer, there is a cassette. So we standardized, we did a lot of experiments, we standardized the cassette, and finally we got something which the flow was right, everything was right, and the next challenge was to develop a scale of the color through which we would make out. So we also got a color chart like this after doing multiple experiments, so that we can do grading of each time we get a response. Each sample is likely to give you a different response. So you will say what is negative, what is positive, and at what range. This kind of color chart also helps us in doing stability studies. For example, if I had a product which was set here, okay, so this is also positive, this is positive, this is positive, this is positive, but if I'm doing stability studies, I cannot say that until here I will be accepting it. No. For a stability study, maybe it is here, 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 that's it. If it goes beyond that, your stability is failed. Okay. This is just for rapid, I'm showing you, because this kit had two components. When it goes to different kind of products, the measurements will change. Okay. Then comes a very important aspect, design verification. So basically, I tell you what is there in the publications is basically nothing but design verification, and it becomes a clinical evidence. So in-house performance evaluations, where you get some clinical specimens, type and specimen stability, analytical sensitivity and specificity, interference and cross-reactivity, precision and accelerated state rate. The outcome, the wrong outcome of this is, many of the research labs say this is my clinical sensitivity based on this work, which is not correct. Okay? So it's part of the verification. Once we have done the verification, I can't even explain this, it's really gone bad. Okay? So then once we have done the verification, we do the design transfer and we do the test batches. Test batches is where we do it at a little larger level, and in the regulation we have to obtain the test licenses from CDSCO. So those have been taken and done, and then we do the performance evaluation. And I'm happy to share that this particular kit, it was submitted to ICMR NIRT, and we got, you can see that accuracy is 98%, sensitivity is 97%, specificity is 100%, and we have recently concluded all this work for, you see this is for TB, this is for rifampicin, this is for isoniazid, and we have a beautiful recommendation, sorry it could read but it's not, that this product can actually now go to National TB Elimination Program. So what I've tried to tell you is a sequence of events in which an in vitro diagnostic product has to be developed or a medical device has to be developed so that we have compliance requirement understood early on, so that when we conclude, we know what we wanted to start with, that this is what regulation would expect from us, and this is where we have reached. So it's a very important and very significant way of doing it. So this is the team which actually did this molecular test development in Hyderabad, and we just celebrated this because a kit has been recommended for national TV program. Of course, it still needs DCGI manufacturing license permissions and all. Thank you, and if there is any question, I would like to. You're welcome. Yeah, he has a question.
Thank you, sir.
You're welcome.
Sir, I have two questions. What is the criteria to be considered a software? It comes under the IVD.
Very good question.
And my second question is, if software comes under the IVD, what is the reference guideline for the validation or qualification?
Beautiful. So there are different kinds of softwares. One is called, if you're using a machine, like for example, you're using a glucometer, it also has a software which is called embedded software, which is also called firmware. It is only to run the machine, gives you a value. Okay. Now, if there is a software which takes values from 10 different tests, okay, and does some diagnosis, what is the intended purpose is not the data analysis. The intended purpose is to diagnose some condition. The data input can come from anywhere. So it has to be an independent standalone software, not the part of any machine, any instrument, of anything. It takes inputs from all possible, like for example, there could be 10 different types of glucometers in the market. So if you take that output into this as an input and then do some, then run your codes and all, and you come out with a diagnosis, then it's a software as a medical device. That's one question, right? Now, once we talk about its validation, so you have to take again design inputs, design and development, user needs, everything you have to run through. Along with this, you also have to run through some of the ISO standards, which talks about which talks about software's safety. Okay, so those additional softwares come into the picture. ISO standards, using them, you have to validate your software that it's performing the same function.
I'm asking because, is it the validation activity is the same as for the pharmaceutical equipment?
Okay, that's a very good question. It's going to help you, and it's very close to my heart because I work on this. Instruments are always qualified. Instruments are not validated ever. The processes are validated. Okay, so which process is validated? Where the output of that process cannot be tested 100%. That is the definition. To make it simple, if you have 100-liter milk, you subject it to a pasteurization condition. So pasteurization is done by a machine. So you qualify the machine that will give you 65-degree temperature consistently for 1 hour. 100 times you do, it will give you the same conditions. That's qualification. Pasteurization is a process. Now it will need several kinds of validations, including microbiology, right? So, and once we do the pasteurization, we cannot test this 100-liter and waste it. It's destructive testing, right? So that's how the validation concepts come into picture. There are two things in validation when it comes to medical devices. One is process validation, that's about manufacturing. Then the product validation is when it goes in the hands of the clinicians. Product validation in medical device and IVD is about its intended use. If the kit, this is a very big challenge in identifying the consultants and actually getting it done.
But then, you know, in case of a university or an institute, I mean, then they need actually a panel of consultants, right? I mean, because
I think for the first year, once it starts rolling out, probably a year later, you will find more and more people talking the same language, more and more people are doing the things right. Yes, initial 1 year is going to be very tough because your entire things have to be channelized through this route. But then the output is going to be very, very good. That I can definitely tell you.
Thank you.
Yeah. Yes, sir.
perform those ISO tests in our lab, okay, any specific test, and show the results, for example, simple cytotoxicity, okay, there is a specific test in ISO, we can perform. Is it valid? Can we go with that? No, it's not, because
laboratory has to be accredited.
Yeah, so that is another bottleneck. Yes. What in the academia we feel that laboratory has to be like accredited by ISO 13485 or maybe 9001. I'm not sure, something like that.
No, I tell you in addition to
how
this.
This can be technical.
Yes, I will tell you in addition to this. I was just working with Professor Bikramjit Basu in ISC Bangalore. So regarding a dental implant. He told me 1 hour data he showed me. He said, Sanjeev, we have done all kind of biocompatibility, this, that, blah, blah, blah. Okay. But what does the regulation want? Regulation does not want biocompatibility to be done on the raw materials. No. If you are taking a raw material, you are subjecting it to a manufacturing process. The biocompatibility should be done at the final packed product. That is why most of the biocompatibility results are rejected. That's one thing. Okay. Second thing is who's doing it. Normally in academia you send it to academic labs. Unless those labs are accredited, these studies cannot be considered acceptable in the regulation. So it has to go to a accredited lab either for 17021 NABL accreditation or OECD accreditation. That's must. If you don't go through that, your biocompatibility studies are good. As I said, clinical evidence, good, no problem. But when it moves from there, no value.
They all can be considered at design input probably?
You have to consider that at the input. Okay. But then, if you are like in the input you say, I will use biocompatible. See, how do you control your biocompatibility? Through the material, through the process, right? So, when you want to control it through the material, that will be one of your checkpoints that whatever material I am going to use, these should also be biocompatible. 80% job done. Now you make sure that the process which you are doing is not increasing this 80%, decreasing this 80% to 40%. That's a 20%, right? That is why the finished product has to be subjected to biocompatibility tests. Yes.
Also, like, what is the requirement for GMP, GLP?
That's a good question. So, who needs GLP? Many people think every manufacturer needs GLP. That's wrong. A GLP is required to a laboratory which is involved into regulatory level testing and has to generate a report for someone else. Unless there is a third party, no GLP test certification is needed. That's the bottom line. Okay. GMP is when you want to run your trial batches. It's good that you do GMP, what Taruna did. She did all her clinical evidence, then she came to us and we did the GMP manufacturing for her kits, which went into validation. So it's good that once you reach to that level, it should be done. But the confusion remains these days, whether you want to remain as an innovator or you want to become a manufacturer or you want to do what. That is a confusion which leads to a lot of problems. Please decide what you want to do with your technology. You want to sell technology, sell it with clinical evidence, that's it. Let other people develop it. Or you complete the development and you graduate as a manufacturer. Decide, because that's how the regulation flows. It's not that everything you will do GMP also, GLP also, this also, that also. It's you're bringing no value because everything has to be repeated, right? So you double expenses, lesser benefits.
Yeah, sir, lucky to have a short time training session when I was there in Tata Steel regarding that hydroxyapatite and collagen validation.
Your name?
Yeah, my name is Dr. Kaushik Kappa.
Achha, you were there?
Yeah, I was there with IIT Kharagpur, so
okay,
that time I took the training. So just I have one small question. If I am developing a buccal patch for the periodontal disorder, whether that will qualify under the drug or it will be a device?
Okay, I think sir, you should be answering this. It's okay, sir. I'm happy. I'm happy, sir. I'm happy.
So I think we can continue with the panel discussion.
Yeah. I think that's nice. Please come. Please come.
Yeah.
So basically, to answer that question, sir, are you answering his question?
Yeah.
Okay. So to answer your question,
my excitement is gone, to be very honest, right? But then, if for the user or for the industry or for the regulatory body, they need it in a round cover slip, and for the 5 years I have done it on a square cover slip, I don't have the energy to do it again on the round cover slip. So if I knew at the very beginning that I have to do it in the round cover slip, that would have saved a lot of time, money, and finally it could have reached somewhere. So I think that bridge is really, really missing.
I think I'll give you one more example. It will probably connect thinking. It's connected. It's a column. So basically, how do we take design inputs? In a COVID time scenario when the hospitals were full with people, and seriousness was different in different patients. They had to be shifted from one area to another area for testing, evaluations, or sometimes not because they were infected patients. But somebody said, I want a portable, what is supporter oxygen, this thing? Portable oxygen meter, right? Ventilator, okay? They said it is important so that we can move all across hospital, this patient with the portable ventilator, okay? Now, how do we take the design inputs? This is just a user need, okay? Design input is now, if you're talking about AIMS, this 2 kilometer area has to be covered, say worst case scenario, okay? 2 kilometer area through several lifts, several corridors, okay? So it could be a physical stability requirement of that portable ventilator. It could be an hour of battery backup which is required, okay? So these are like two things if you want to talk about. When you move patients in such a scenario, the breathing also increases. So what is going to be the breathing rate? What is going to be the total oxygen required? Okay, once you select all, these are called product requirements. Now product requirements are converted into a design input. What is design input? Okay, we are going to have a battery now, which is going to give us minimum 90 minutes backup. It has to be a lithium battery rechargeable. Product specification. Okay, what is the regulatory requirement? Next point. Are the batteries controlled under some regulation? Yes, bring that regulation. Because this battery which I am going to select has to comply with that regulation, then only my portable ventilator will get approval. Otherwise it will not get approval, right? Now once it is moving, it has to be electrically safe. It has to be magnetically safe. There is a standard for everything, right? If you don't consider those in the beginning, you don't test the final product in totality. That's the benefit and advantage of having user needs getting converted into product requirements, converting into design inputs, converting into the final target specification. Then you start working. You know what happens in industry also, people join and they are put to work next day because HR says we can't waste time. Well, you have to do the good training so that they give you the very nice output. That's very, in your labs, how many times do you really, you go and see whether the pH meter is rightly calibrated? It's not part of your way of working, right? That's the point which is important. And when the regulator wants it, he'll say, Acha, this is your pH data, please show me your calibration record. If you're not showing calibration record, that becomes unacceptable. Now just imagine you are doing 2 years of work, you're checking pH, but you're not able to show the calibration record. Your submission is going for a 6.
Also, maybe many of the times we actually did the calibration,
but you don't have record. You don't have record. Yes, and this is what is technically lacking in the entire academia space.
Let me just add one to that. As you were talking about the the user need and the specification, something I mean obviously without all this knowledge, but just from the intuitive one, something I keep on telling to the PhD students, in the research committee I am in, or I don't know, some of your poster in the, yesterday I was, going through, and they, when people talk about that, we have got this optimum result or the optimized result. I ask them, how do you know this is optimum?
Correct.
You have seen, okay, you vary NaOH, you got a better result, but what is your requirement? What is your specification? What is your target? And I see that many of the times we really don't know. I mean, not just the student, we don't know that what is the
Yes,
target.
yes,
I
I
think some of
agree.
they are already smiling because I asked them these questions and you know, yeah. So.
You were saying something, Karuna, in between?
Through some quantifications, the software actually detects that this is the kind of value they have created. So alternatives they get, and from there they decide the optimum results. Optimization.
So the optimization, I mean, first of all, optimization is a complete mathematical problem, right? That needs to be done using DOE, as you were talking about. And another is that the optimization, I think, I mean, you can correct me if I am wrong, like the optimized result and the user need result, they may not be the same, right? I mean, optimization, you're optimizing based on what? Are
Absolutely.
you optimizing the money? Are you optimizing
Absolutely.
the performance? Or something?
Yeah, I just wanted to say, Neetu was saying that, you know, exposure is needed and right from the beginning we should know. This is one level of difficulty. But I have seen he has been working with the industry and being his life partner and sharing his, you know, every evening we share whatever we learn through the day. So I understand even the industry is not able to follow the regulations properly. Academia is going to have a real tough time. मैंने खुद काम किया है उनके साथ. I almost cried when I worked with him. So it is not going to be easy. So that is another different level of difficulty you are going to get.
So, in fact, I would share my very recent experience with this company in Spain, Bflow, and we have had arguments where I really don't understand and I get annoyed because they keep asking me about how many times we've repeated the experiments. The replicates, the technical replicates, the pH calibration and all those kind of things. And you know, I tell them that, what do you mean we have done? You know, they're not criticizing. But it is a difficult thing for me to understand. See, and you know, the reason I'm talking about it is, in India, we kind of escape from not giving things in the market because we say we don't have the ecosystem. The industry is not coming forward and taking our technologies, right? This is a very standard thing you will hear from many of us. I have seen the system in the US also. Maybe there are takers, maybe the industry is there because they've already invested into consultants like this. They've already, they say, the PIs basically just transfer it to those people. They say that, you know, this is kind of what we're talking about. We don't want to understand anything. You do it, you know, they literally give the input that he was talking. Now it all makes sense to me.
You will all be talking about this now.
But in India, our industry has spent money for a particular product. They do not have funds to go beyond. So for example, we're talking about 3D. All the pharma companies have invested money in 2D. They have set up everything with 2D. I go to them and say your 2D is waste of money or waste of time or whatever. My 3D system is better. They're not going to change that system overnight and come to you unless I give them convincing data that he's talking about. So if I really want that industry to come and join me in my journey, I think now I should be giving them this data.
Absolutely.
May not be to the regulators. They will take care of the regulators, but even, so it's like, you know, a college kid coming to us. We see that a lot in IITs. The campus kids, you know, in their hostels, they would do some experiment. They would bring something to us and say, ma'am, this is what we've done. I asked them, where have you done this? Ma'am, hostel ke room mein. I was like, room में ये experiment कैसे? मैम एक mixer लिया, grinder लिया, उसमें ये डाला, ये किया, वो किया, and then we're like, wait a minute, this is not correct. I don't think you've done anything. You come to the lab. That is exactly what the industry is telling us when we show them data. Although we show them all our publications and patents and all that, but that has been validated scientifically. The science is there, but whether it is going to work, as you were saying, that's a different thing. So I think we need to cover a lot of journey. After hearing his talk, I feel like, you know, I knew it. I knew that there is some problem, but what that problem was, I was not able to locate because I was having, so, you know, I did try a lot of things, and then finally I got a company who was interested in us and we started working together, but then we're not able to talk. It's very, very frustrating.
You're not in sync, yes.
We are not in sync. I don't understand what they're asking me and I get very annoyed. I feel like they're doubting my data.
These are trust issues.
Exactly. These are not requirements. So I have had this. I was like, why is he asking me this? And then like, but he won't, this cannot be trust because he has been with us and done those things. So it can't be trust. So I was a little confused, but now it all makes sense.
Yes, yes, yes. It goes much beyond, you know. Sir.
Coming back to medical devices,
Yeah.
with the government now issuing the list of things covered under medical devices, and also government notifying the names of labs which are included, approved for the testing. So I could see that there are not many labs, and most of the labs are just mainly doing few of those requirements. So how about the tests which are not done in any of the labs for those medical devices?
So this is a big challenge, sir, and I would just say that just the last week, our Honorable Minister, Mr. Piyush Goyal, he has had a cross-functional two-day seminar only on finding out the gaps in the testing systems. And they are talking about laboratories, they are talking about funding them, they are talking about expediting their accreditations and other things. And this is just a continuous process. Industry is so happy, they're so happy that this is being taken up. Okay, like in COVID times, there was a war room. That is why we found laboratories everywhere very quickly. And this war room had the Department of Pharmaceutical, ICMR, CDSCO, Finance Ministry, all approvals were given in one go. That is why we could see COVID products moving. But it has been taken very seriously. Ministry of Textile has been roped in for textile related medical devices. Steel has been roped in for steel related materials. And it's happening massive, sir. And I tell you that even CDSCO has been granted more than 2,000 crores for upgrade. It's not less money. 2,000 crores means infrastructure, people, so much of it, everything is automatic. I just submitted one of my clients' document to US FDA. I submitted, uploaded this. It came back in 24 hours that your file names are not correct. Can you imagine? The file names are not correct. Okay? I was totally taken aback. File names are not correct. I downloaded everything and I said, now what I do, I have done corrections. So again went back to the site, it says we have a file name validation tool available online. Download the tool, subject your file to it. If it says good to go ahead, submit it. Just imagine where we have to reach. Okay? So that is there, and more laboratories I think are going to come, sir. And we're looking forward that we started CMDT and here. That's running a little slow, but we should be able to do it.
Hopefully.
Yes. So just to add to both of you's learning that NIPER Mohali is going to have a medical device testing lab which will test combination products. Combination means which have drug into it and a vehicle into it. So this lab is going to specialize in that because these people have super ability to do drug testing. So medical they will add. And there is a small committee and 3 of us are on that committee and we are pushing it under Ministry of Health and Welfare again. Oh, chemical and fertilizers probably.
Yes. Apart from that, we are also running medical device courses.
Yes.
Madam is doing that, and some of our faculty colleagues are running that course.
Correct.
Master's course in medical devices.
It's good. Yeah.
Just for your simplicity, what is a combination product? The audience, yes, what is a combination product? Combination medical device product.
You will get an extra ice cream in the dinner.
No, it is just to, make your eyes wide open. It has a drug component and a device component. Simple being insulin pen. I hope everybody would have seen an insulin pen. Fixed dose, multiple dose, titrated dose, yes? Or nowadays we also see some medicated surgical dressings, isn't it? A lot of...
Yeah, even drug-eluting stent is a combination.
Drug-eluting stent, that's a very classical example, and I think everybody knows. We heard few years back 70% price reduction in drug-eluting stents by CDSCO. No interest. I don't see the smiles in the audience.
Any medical device or stent is coated with the drug, and primary intended action of the
stent only. Is it comes under the drug combination, drug medical device combination, or only medical device? Very, very good question. Very, very good. I have the mic, sir. So this is a borderline case. This is a borderline case. But here now, we'll have to see, like you did the biocompatibility of the drug stent material, or stent material, right? But now if you're coating it with something, now your biocompatibility requirements will change. Then the stability of the coating, okay? But it may not come, it is not a combination device, I can tell you that. Because like his good friend is working on, no, this. No. I asked you, ceramics, bioceramics, hydroxyapatite, okay? Hydroxyapatite is coated probably on every medical device so that there is enough osteointegration, okay? But it does not make it a drug combination. It will be considered as
Yes.
a medical device. Yes. We have to apply for that.
Yes, but if you now bring antibiotics, you say it gives stability also, but you are claiming that it will be saving it from the infections also.
Yes,
then it will become a.
In the guideline that if the drug's primary intent of drug action is pharmacological action,
Correct.
then it will be considered as a drug.
That is the definition. So you know that Celestin story, it's the same story.
You mean Jayesh?
That is the
No.
When will our
Pudar.
Achha,
Kingshuk Pudar.
When we expect our Indian regulations will be in complete defined way for the medical devices?
Why do you think it is not yet?
Because where I was referring, it was mentioned that for the quality assurance, quality performance study, we have to follow the ISO guideline. So ISO so and so guideline. For all these kind of guidelines, they have just referred the WHO and ISO. If we have our own.
Okay. So you're coming back to the title of my talk, basically standards and monographs, right? Correct. You are absolutely right.
Many of them mention that for the particular evaluation, just refer ISO.
Yeah. So have you seen the vacutainers, the blood collection tubes?
Yes.
Do you know that India had a standard for vacutainers in 1984? Does anyone know it? No. Where does it come from? ISO standard has come much later. So it's there. Many standards are there we didn't know. Okay. For example, ventilators. if not the ICU ventilator, there were standards for ambulatory ventilators. There are 3 different kinds of ventilators. The entire halla-bolo during COVID-19 was about ICU ventilators, not about ambulatory ventilators or bedside oxygen ventilators. There was no problem in that. It was only ICU ventilators, right? Like this, there are a lot of standards, but yes, medical device has been primarily a field of outside India. Even today, we are importing 80% high-end medical devices. So if there is no product manufacturing in India, how will the standards come up? Okay, and there is another requirement, I tell you. ISO standards are translated verbatim as IS standards. If there is a Bureau of Indian Standard which is not existing in EU and they need it, they will translate this into an ISO standard. It's a global activity signed and agreed between different governments. So it works. That is why ISO standards are acceptable. they read ISO. तो अभी CDSCO के साथ BIS की वेबसाइट भी चेक करेंगे. And they have constituted recently a lot of committees. More and more standards will come. Yes, of course.
Yeah.
I mean, you should not feel so, gleamy faces as we have moved, with such great development over past 2 decades in clinical research and clinical trials. We will surely go ahead and develop more and more standards, and we'll get ourselves equipped as well as knowledgeable enough. As we'll just practice, this is my belief, as we'll just practice, we'll go in the flow and we'll start using these technologies, and we'll go, it will become a habit. Like earlier, getting an ethics approval was you considered to be a nightmare, literally a nightmare. Oh, when I had put my DCGI application in 2007, I was pissed off. When I took my first consent form signed by the patient in 2005, you can't imagine how much butterfly I was having in my stomach. So over the years, it has become a habit. Similarly, I feel that medical devices testing, adopting ISO standards, going through the BIS sites, following all the procedures, it will become a habit.
And exposure also,
Yes, of
earlier in
course.
Drugs and Cosmetic Act, there was Schedule Y for clinical trials. Now that has been split out. For cosmetics, they have made another rules. For medical device, they have created separate rules. And just a point of, just point of addition here is that if you have global market in your mind, when you're writing your protocols, clinical protocols, we also consider ISO 14155. Now, if you want to sell any of your product in EU and you say, we have done the clinical studies and you don't even mention 14155, namaste. You don't take it, right? So all your efforts of 2 years have gone vain. So this is where the regulatory strategies are very, very important. I have been telling all incubator heads, get it right please, but seems to be everyone wants everything for free. That is my limitation. Gola घास से यारी करेगा तो खाएगा क्या? So this is what is something which is very important, that every incubator needs it. I tell you, every incubator.
I think those kind of investment should be done in every institute.
Absolutely.
Right? The patent law, of that. I think that is coming now slowly.
It has become better, at least in the institutes we've seen that patent-wise they have become better. Still not as good as CSAR places and other places, but slightly better. But this is definitely missing. We have, in fact, IT Delhi also got something in medical devices. We are one of those 5 IITs chosen to manufacture medical devices. We are setting up a big lab. It's called M Pragati. And I'm going to go back and again talk about this because it is going to be important. You can't have a manufacturing setup without these things. And in fact, this can be like a certification program that I think we should give to anyone who's coming to use that lab, because it's impossible for people to get space in incubators and things like that without knowing what they're doing. So Exactly. That's what I'm saying. So like he's talking about incubators, right? IT Delhi is all about incubation. Sorry to interrupt. We have incubators. We have a lot of people working on medical devices who are being incubated, but we do not have these kind of knowledge. So they are working over the year, and that is why we're not seeing anything coming in the market, because they fill up, those documents and really long dossiers, and they go and they are rejected, and then the person
I guess
loses.
as you was talking about the pH meter, right? So for example, all the various central facilities that we as well as those incubators they're using, but then I guess even for all those facilities there also needs to be a log of the data record, right? But that's not there, right?
So I will give you last flavor of this conversation. Probably we are going too long. In one of the companies, R&D would do some projects. They will come out with the product. Coming out with the product just does not mean the product. It comes with the process. It comes with a lot of documents. So they will submit the documents for the review. That's part of the design verification. So my requirement as a quality head and regulatory head was, whatever you have concluded for any of your parameter, take me to your raw data. If I don't see this conclusion in your raw data, it's unacceptable to me. And today you go, I left that company 10 years ago, you go, they're still working in the same manner. That's the beauty of it. So you go, like for quality people, the green color pen is very important. We can't use red, we can't use black, we can either use blue or green. Okay. So when I am doing this verification, I'll go to the raw data, I will sign it, okay, and take that page number into what I am reviewing and cross-connecting it. So 10 years later, if someone wants to see where does it come from, it comes from that register, that page, where the green signatures are there. That's the connectivity. And that's what regulation wants. Thank you, sir.
Thank you very much. Thank you, everyone. So we can, I think, close this panel discussion now. And we thank all our
Hello. So one announcement for the students. All the students are requested to please collect their certificate from the Friday evening to Saturday evening at the certificate committee desk in the convention center lawn, and also check your poster, oral and attendee number from the board placed at the certificate committee desk. Thank you.